International Symposium on Agricultural Transformation and Biotech Crops in Africa. We'll deliberate on what agricultural sector stakeholders need to do differently to ensure expanded farmer access to crops produced using new breeding techniques. Symposium participants will deliberate on how genome editing tools like zinc finger, nucleus, talents, CRISPR-Cas, among others, can be used to improve crops for the benefit of African farmers. Participants will also deliberate on how genetically modified technology has been deployed on the African continent for the benefit of farmers, its impact so far, and its prospects. Also, participants will deliberate on the yet-to-be-realized potential that genome editing holds for the ongoing efforts to ensure Africa feeds itself. Good morning, ladies and gentlemen, and welcome to this third day of our activities at the West Africa Center for Crop Improvement. Soyez les bienvenus. Mes frères et mes sœurs de la région francophonie, on est vraiment fiers de vous avoir ici et on va essayer de faire ce qu'il faut pour to make you happy. C'est déjà bon, n'est-ce pas? All right. So, distinguished ladies and gentlemen, we want to take a few housekeeping rules as we get today's project underway. So, first of all, for those of us who may not be very familiar with this building, if you need to use the washroom, you step out of the door out of the second door and take your left. The washrooms are there. In the unlikely event that there is a fire alarm, we all proceed through the second door, and let's use the stairs, not the escalator, and walk gently downstairs and assemble in front of the building. We pray there will be no fire in this building today. We'll be fine. Then also, for those of us who may not have space in here, we have an auditorium downstairs fitted with a projector and a monitor, so you can go down if you don't have a space here. Go down, take a seat there, and watch comfortably. Then also, we need to make a quick announcement about how we're going to use our time here today. We want to do everything and close from here latest by 3 p.m. We'll have some breaks in between. We are going to insist on the right use of time. So, when you have the opportunity to either address the gathering or ask a question or speak to any issue, we'll be timing you. For those who will be having lengthier spurts of time to speak, as you do a couple of minutes, we'll be prompting you. There'll be somebody at the back who will flash a card at you to say 7 minutes more or 5 minutes more, and we want to go straightly by the time so that we don't overstay our welcome here today. Distinguished ladies and gentlemen, having said that, and if all hands are clear, may I now have the privilege to invite the founding director of the West African Center for Crop Improvement in the University of Ghana, Professor Eric Irechi Dankwa, who is also the current Africa Food Prize laureate. Please, ladies and gentlemen, put our hands together as we invite Professor Dankwa to come.
Good morning. Good morning. Someone was asking me, he thought I'll be in bed. Yesterday night was another story. I've been in this university for close to 32 years. Since that auditorium was built, I've never seen an overflow. in that building. So let's give the West Africa Center for Crop Improvement a round of applause. You saw how our Vice Chancellor was dancing. We made history. We made history. When I was growing up, I played soccer, football, and I was a great footballer. I played for one of the greatest Coles teams in the country. And in football, when you play away matches, you get scared. Over the past 2 days, we've been playing away matches at Ise and and at the at the City Conference Center. Now we are home. So this is a home match. And I believe today is going to be the most fascinating day. We were expecting a couple of others to join us before the start. They haven't come. And Mr. MC, I'm the only one who does not have time to speak because 2 speakers are not here, so I have the privilege to speak for as long as I can. Thank you, Mr. MC. Country Lead Alliance for Science, Mr. Joseph Gapo, are you here? Keynote speaker, Dr. Linda Tripathi. Friends from the media. I wanted to say friends of the media because I'm becoming a media person. Distinguished ladies and gentlemen, all protocols observed. A very warm welcome to this international symposium on agricultural transformation and biotech crops in Africa, with a focus on new tools, technologies, and methods. For me, it is an honor to have such distinguished personalities. If you have looked around this auditorium, you find people from across the globe over here. It's an honor for me to have all of you gather here to discuss and explore the fascinating field of agricultural biotechnology and genome editing. When you look around in today's world, people are suffering all over. This week I have said that some of us have never experienced hunger. But there is hunger everywhere. There is crisis around the world. Climate is changing. The rains don't come. The floods come at the wrong time. Crops fail. People suffer. Population is growing. In Ghana alone, we heard that we would have over 5 million additional mouths to feed in the next 7 years. These are troubling challenges. And when you look at crisis everywhere, the impact of the Ukraine, Russia, what is it? Was it a war? Is it a war? I once said that why should 2 sovereign countries exercising their sovereign right to do what they want to do affect another country? It's a shame for Africa that we are hungry and we look up to Ukraine and Russia to bring us food to eat. In today's world, where the challenges are becoming too serious, the power of science and technology can offer sustainable solutions. And I wish my Honorable Deputy Minister for Agriculture was here because I wanted to share a few words with him. Plant biotechnology and genome editing offer tremendous opportunities to address these challenges by enhancing crop productivity, improving disease resistance, and ensuring environmental sustainability. I believe that this symposium serves as a platform for knowledge exchange, collaboration, and the sharing of groundbreaking research and innovations. What we want to do today, and our MC is beyond an MC. He has, in fact, stated the purpose of this meeting. What we want to do is to delve into the frontiers of plant biotechnology, unlocking the mysteries of plant genomes and leveraging cutting-edge tools like CRISPR-Cas9 and other technologies. I believe Nigel Taylor is going to unlock some of the things that they are doing at the Danforth Center. A few weeks ago, I was with him when he started describing what was coming out of their research. I was so excited, and I was happy that within a minute I could convince him to join us in this meeting. Thanks, Nigel. I hope that the presentations will cover a wide range of topics, and looking at the program, I'm excited, including the latest advances in genome editing techniques, the application of plant biotechnology in developing, if you like, resilient crops. And also, we hope that there are lessons we can learn from the work going on in Latin America. I believe it has implications for what Africa should do. I also believe that this gathering offers us the unique opportunity for networking and forging new collaborations. And I would like to encourage all of us to have very fruitful discussions. We need to exchange ideas. And of course, I believe that there are going to be some bonds today which will be long-lasting. By pooling our knowledge and expertise, we can pave the way for groundbreaking discoveries and innovations that will shape the future of agriculture and contribute to sustainable development in Africa. I would like to extend my heartfelt gratitude to the organizing committee. Joseph, thank you very much. We had to work together as a strong team, the WACCI team that worked with the Ghana Alliance for Science. Thank you very much for all of the hard work. I also would like to thank, I was hoping that Dr. Sheila Ochuboju will be here. I want to thank the Alliance for Science, BTI, Cornell University for the sponsorship that has allowed this meeting to become a reality. Joseph and your team, your dedication to the hard work is truly commendable, and we are grateful for your efforts in creating this platform for scientific exchange. I believe we are seeing what is happening in Ghana today at the Supreme Court. Is it great news? Are people going to be quiet forever? I also like to express my sincere appreciation to each and every one of you. I believe a number of you are tired and wish you could rest the third day, and then those of you who are seeing a haven in Ghana, Accra, want to look around and do some shopping. We are sorry that we didn't free some time for you to do that. Apologies from me. But your presence here today reflects your commitment to advancing the frontiers of science and your passion for making a positive impact in our world. I believe together we can explore new horizons, which is important. We can push the boundaries of knowledge and unlock the immense potential of plant biotechnology and genome editing. So once again, I would like to warmly welcome all of you to this symposium on agricultural biotechnology and biotech crops. Let us embark on this journey of discovery and collaboration, and I hope this event becomes the catalyst for groundbreaking advancements that will shape the future of agriculture and secure a sustainable future for generations yet to come. Before I sit down, Alliance for Science Ghana, I would like to propose that this WACCI Alliance for Science partnership be instituted into an annual convening at the West Africa Center for Crop Improvement for conversations on new tools, technologies, and methods for transforming agriculture in Africa. I further propose that it coincides with World Hunger Day. You saw what we did on World Hunger Day. I believe we can create a new platform that will bring the best from all over the world to convene at the one and only West Africa Center for Crop Improvement. And I can see Giles going back and booking his ticket for next year, May, to join us in this first conversation after this year. I saw his clap was the loudest. It is my hope that in the fullness of time, this convening will grow into one of the biggest platforms in Africa. And I can see Lina jealous, one of the big platforms in Africa for conversations on plant biotechnologies and genome editing in Africa. And of course, WACCI, we have begun to do wonders. We have started conversations with UC Davis, with the UC Berkeley, and also with the Danforth Center about how we can create one of the largest platforms for genome editing in all over sub-Saharan Africa. I believe we are doing that, and we want to compete with, for example, UM6P, what we saw recently when we visited, having one of the largest platforms for phenotyping in the world. I believe is a big plus for UM6P. And I hope that we can transform this university into a platform for good science and technology so we can attract the best in the world to come and work with us. I would like to see ideas birthed on African soil, turned into innovations in Africa for Africa. So this is a new philosophy. We want the ideas. We wouldn't go to San Diego to birth those ideas. We'll come to Africa and birth the ideas in Africa for Africa. And I believe that when people like Giles, Rita Moore, Ronnie Kaufman, what's his name? My good friend Nigel Taylor, when they move from where they are, Evans Laguda, when they move from where they are, from Australia, from Danforth, from Cornell, from Illinois, from the ITA, when they come to the West Africa Center for Crop Improvement and we brainstorm together, we turn our ideas into innovations in Africa for Africa. I believe that you see that, yes, we will be the originators. They would have contributed to the conversations, and together we'll be going forward. Distinguished ladies and gentlemen, I gladly open this important symposium, and I pray that we all find it very fruitful. Thank you very much.
Thank you very much, Prof. You actually did less time than I thought. You gave me 5 minutes extra to work with. Thank you very much. So the clarion call is very clear. Let's work towards birthing ideas here in Africa for Africa. Thank you very much. We're going to take a very short video from, what's the minute, am I ready? Yeah, from Dr. Sheila Ochubogu, who is unable to be with us in person here at this moment. So please, let's have that video.
Hello and welcome to this International Symposium on Agricultural Transformation and Biotech Crops in Africa. I would have loved to be with you today in person, but unfortunately I've been called away on urgent business. Our team, Alliance for Science team, came to celebrate on Sunday 28th of May the launch of our National Farmers Day campaign. We launched the campaign to get farmers recognized and National Farmers Day to be instituted across the global south, because we wanted to learn from the example of Ghana, which actually instituted a National Farmers Day in 1985. And it did so because Ghana was one of the first African countries that was really working towards cutting hunger by changing economic policies and investing more in agriculture. That was in 1985. And by 2011, the former President Kufuor won the World Food Prize because he was one of the first leaders in an African country that had reached halfway towards SDG 2, zero hunger. It was a great achievement. Now we find ourselves in a world of increasing vulnerabilities. And those vulnerabilities are evident all around us in terms of climate change, in terms of conflict, in terms of higher prices for inputs. Everywhere our agricultural systems are being challenged and strained. But one thing we can rely on is that if we give farmers good seeds, if we invest in good inputs, there will be an agricultural transformation, and we can see the possibility of ending hunger in our lifetime. So this symposium here in Ghana is to celebrate the fact that on this continent, we have the innovators, we have the innovations, and we have the strength of will, because we're living amidst the challenges to want to achieve zero hunger. All of you who are gathered here today, and all of you who are going to make contribution, will take us a little step further towards realizing that global aspiration. You'll be sharing knowledge about technologies, techniques, novel methods, and you'll be doing it in a way that we can hear each other, learn from each other, and as a continent of 54 countries, different agroecological zones, different economic and demographic challenges, we can hear each other. We can say our brother from the east, north, south, or west is doing something that perhaps I can apply here. And our innovations and our technologies can accelerate us towards our stated goals. I hope the whole world will be able to hear some of the conversations and discussions that are happening in this room, and to learn from you, and to reach out to you to help on the path towards zero hunger. Thank you very much.
I have just 2.5 pages, so I'll be done very soon. But Professor Dankwa, the Alliance for Science team, all the WACCI alumni, ladies and gentlemen, it's an honor to be here with you to celebrate the homecoming event of WACCI and also your 16th anniversary. Well, also, we have a number of guests joining us from across the world who are streaming this particular event live on the Alliance for Science social media platforms, including YouTube and Facebook. We say many thanks to all of you for joining us. I see a lot of old friends here, people that we've been involved in the biotech space for many years. But then I see a lot more new faces here, even as we converge today. At Alliance for Science Ghana, we've repeatedly been having conversations about how most of the times, the majority of the audience that we engage when we hold our meetings, when we hold our conferences, when we hold our symposia, are the same people that we've been meeting over and over again. So the last 6-7 years, when you walk into one meeting or the other, then the same batch of people, including the journalists who cover our events. We've seen situations where the same group of people keep coming back again. But through our association with WACCI, we are opening up to a whole new audience. We are opening up to a lot of accomplished alumni from WACCI who are working at various national research organizations across Africa. So for us, this is an opportunity that we, the team at Alliance for Science Ghana, are very much grateful for. Let me pay tribute to my mentor, Professor Eric Dankwa, not only for building this great institution called WACCI, but also making time to mentor a lot of other young people right here at the WACCI institution and even across the world, and including the team at Alliance for Science Ghana. So I remember emailing Professor Eric Dankwa some few months ago and telling him that Alliance for Science would be interested in coming up with a symposium of a sort that then tries to then broaden the reach of the entire biotech conversation beyond the audiences that we've always known. And interesting enough, just when we prompted him about this plan that we had, his quick response was, let's do this. And he said we could partner and get this done during the WACCI homecoming. He very much embraced the idea with very open arms. But you all know him, he's a very tough guy. He doesn't just take in everything hook, line, and sinker. He picked up the concept notes, he virtually tore it apart with very constructive feedback. And when we sat down to revise the concept notes, all that I had to do was to click Control-A, delete, and start it all over again. So, Prof, thank you very much for your mentorship and your guidance and your support all through these processes. Well, you've spent a lot of time in the academia space, and you would have a few more years to then still be in the academia space and even beyond. But then we are very much clear in our mind that the legacy that you've built and the visions that you've inspired would live for centuries to come. So we're very much grateful to you, Prof. So we say thanks very much for broadening your shoulders and allowing others to climb on that and getting things done. As we would usually say, may your valleys never run dry of flowing water, and may all things that you touch continue to flourish, Prof. Thank you very much once again. And congratulations to the team at WACCI for 16 years of excellence and for setting the pace for other academic institutions to follow. You, the folks at WACCI, make all of us proud. So, Alliance for Science Ghana is an institution that advocates for the prioritization of science when it comes to national development generally. I was first introduced to the biotech advocacy space specifically somewhere in 2009 as a first-year BSc in agricultural biotechnology student at the Kwame Nkrumah University of Science and Technology, sitting in the class of Professor Richard Akroma and others. And I look back 14 years on, and it's amazing how so much has changed in the space over a decade and a half or so. Those days, majority of the players in the biotech advocacy space were scientists and professors like Professor Walter Al-Hassan, who is with us here, Dr. Mumuni Al-Hassan, Dr. Tukule, and others. Scientists, professors, matured folks. But today we see a lot of young people in the space. And a few names come to mind. Dr. Jerry Boyene, who is with us here, Dr. Daniel Osei-Fosu, young people who have also gotten into the biotech space and are not just doing the science, but are also doing the advocacy. That's something that we find interesting, the change that we've seen over the last few years as far as the biotech advocacy space is concerned. That's not the only difference that we've seen over the last decade and a half. I recall that those days, the conversation around GMO was virtually a very static one. We kept on having debates in my class, for example, as a BSc Agricultural Biotechnology student, making the point that, we need GMOs, otherwise then Africa would go hungry. And that was the basis for which, in my class, when we debated biotechnology, we insisted on the need for technologies like this to transform agriculture. But then today, we've expanded the conversation. We're seeing a situation where there's a lot of value-based communication approaches in the general conversation when it comes to GMOs and biotech. This, again, is a generational shift that we find very much interesting. And so today, that is something I would want to then encourage all of us to, again, be interested in doing. When it comes to the conversation around biotech, we've seen situations where a lot of others have ventured into the space. Today, it's not just the scientists. We've had lawyers venture into the space. Pablo Orozco is a lawyer who's been involved in the biotech advocacy space for a while. We've seen journalists venture into the space. I'm not one of those journalists, though, although I'm a practicing journalist myself. Now I probably would call myself other things than a journalist. We've seen situations where the conversation has gone beyond just the academic papers when it comes to science and biotech advocacy, and a lot of it is happening on social media and even in mainstream media. We as a science advocacy organization are hoping that we continue to see more of that, where those of us who are scientists in the space are stepping out there and explaining the science and advocating the science for better days. One final point that I would round up before I get off the podium. Today, we need all hands on deck to deal with the food security situation that we have in Africa and beyond. Statistics from the United Nations Food and Agricultural Organization estimate that 20.2% of the population in Africa faces food insecurity and hunger. When you compare the figures, in Africa we have 20.2% of the population. In Asia, it's around 9.6%. In the Latin American area, it's around 8.6%. In the Oceania region, it's around 5.8%. And even in the North American and also the European area, it's around 2.5%. What this means is that in Africa, we have about 278 million people who are struggling with food insecurity challenges. And that means that particularly in Africa, we need all the necessary tools that can actually help make a difference as far as improving crop production is concerned. That is why it's kind of heartbreaking when you see that here in Africa, we have a situation where we are not seeing the necessary investment being made in science and technology, and particularly plant breeding tools and tools like biotechnology. We've been doing an analysis of the 2023 budget that the Minister of Finance sent to Parliament at the end of last year at Alliance for Science Ghana. And we found very interesting details in there. We found that with the budget that Parliament approved last year for this year, there is the situation where the Ministry of Science, Technology and Innovation, which is supposed to be the engine institution for Ghana's technological drive, is getting a budgetary allocation of somewhere around 302 million Ghana cedis, as against a general budgetary appropriation of 227.8 billion cedis. So we're seeing a situation where the ministry, which is supposed to be the engine of science, technology, and innovation, is getting just 0.1% of the total budgetary allocation. That's certainly not good enough. There's been the advocacy at the level of the African Union for at least 1% to be invested in science, technology, and innovation. And it's about time that all of us in the science space then make the necessary efforts to then urge our governments to then meet these targets and ensure that there is better investment in science, technology, and innovation for the benefit of the general populace. As I conclude, I would want to thank Dr. Sheila Ochubuju and the Alliance for Science Global South team for being here and for supporting this event, as Prof. Dankwa rightly indicated. We thank the team at Alliance for Science Ghana as well for all the support. We look forward to very exciting deliberations all day. At the end of today, we will issue a communique on pathways to making Africa food sufficient based on what will be said at today's symposium. And this document will make recommendations that donor partners, universities, research institutions, industry, farmers, scientists, and other agricultural sector stakeholders could institute to enhance access to made-in-biotech crops. I say we're hoping for very insightful deliberations all day today. Thank you all very much.
Thank you, my friend Joseph. I was going to stand here to wait for you, but when you said you had only 2.5 pages, I advised myself. There is an inherent danger in hearing these numbers over and over and over. The danger is that at some point we become numb to it. We think, oh, it's the same thing we have heard all these years. Nothing is changing, so we don't seem scared anymore. Well, I am. Each time I hear these numbers, I'm scared. There's something that must be done. The call to transforming Africa's agriculture using biotech we cannot say it enough. Distinguished ladies and gentlemen, I'm going to invite the Eastern Africa Director of the International Institute of Tropical Agriculture, IITA, and leader of Biotechnology Program, Dr. Lina Tripathi, to come give us the keynote address, after which we'll have an opportunity to ask a couple of questions, and then we'll take a short break. If our hands are clear, please, let's put our hands together as we invite Dr. Tripathi.
Good morning, everyone. And first of all, I would like to thank Professor Eric Dankwa and Joseph for inviting me as keynote speaker, and so that I can share some of my views. So I'm going to talk about how we can transform Africa's agriculture using biotechnology. So can we switch off the lights here? Actually, this slide is not new to many of you. You all know that, the major global challenges in agriculture are there's the rapid increasing global population. So you see the population is increasing. Already billions of people are going hungry. So now it's increasing. But the challenge is more in Africa, because in Africa by 2050, the prediction is the population will be double. As you can see from this graph. So in order to feed that increasing population, we need almost like about 60% more food. And we cannot be actually only targeting only the few cereal crops. Actually, if we talk about Africa, we have to tackle all the staple food crops grown in Africa. And on top of that, we have another challenge of the climate change. We already started seeing the negative impact of the climate change in several parts of the world, not only on the plants, but also on the pathogen and pest, which affects the plant as well. So when it comes to the agriculture in Africa, actually more than 60% of the population is in sub-Saharan Africa is actually smallholder farmers. So about 23% of sub-Saharan Africa's GDP comes from agriculture. And again, not only that few cereal crop, but also other like, the root and tuber crops, but also other cereal crops are very important for Africa. But it's still, the Africa's full agriculture potential remains untapped. Africa has the potential to produce 2 to 3 times more cereals and other staple food crops, and which should be able to add actually 20% more to the current output globally, not only for Africa. So in order to get actually the agriculture potential, it's not only the biotechnological tools what we need a lot of other things as well. So the major important thing is that we need access to the seeds of the improved varieties and fertilizers, and that's where actually biotech plays very important role. Also the access to the modern farming technologies and irrigation, improved soils, also the research and development, which WACCI is doing very well on the continent, extension services, and also access to the market. We need all of these in order to transform agriculture in Africa. So in order to get the sustainable agriculture, we need to think about how to close this yield gap. So if everything is very good, you have all the technologies you need, including the biotechnology, very good soil, everything, then this is the potential production. But that's not always the scenario. There are the limiting factors. So then this is the attainable production in that case. But then there are again the pests and diseases, which again reduce the production, and that's where the yield gaps comes from. So this is the actual and this is the potential. And it doesn't even stop here, because even there are post-harvest losses as well after that. So we need to find out the innovative approaches combined with the traditional knowledge to increase the productivity in order to close this yield gap, and also to enhance nutrition and build the climate resilience. So in nutshell, what I'm trying to say is that with increasing demand for food and limited resources, we need better and more efficient ways to produce food, and we have to utilize all the tools available in our toolbox, including the new breeding tools, such as the transgenic and the genome editing. When we talk about the genetic modification, actually, basically, a lot of benefits has already been seen, because this is the technology which has been over the decades there. So, you have seen a lot of contribution to the food security, sustainability, and the climate change solution using the genetically modified crops. But still, these crops are actually limited to only for few crops, mainly like, maize, soybean, canola, and then few more crops coming up recently, and cotton definitely. But then, when we talk about in Africa, actually, the most important achievement is the BT cowpea, which has been the first food crop released in Nigeria, and also now coming to Ghana after the cotton and definitely maize, and then in Philippines, the golden rice, which has a lot of promises there. So if we talk about the cultivation of the GM crops, there are more than 29 countries which is planting this crop for ages. But then when we come to Africa, actually in 2019, the number of countries cultivating GM crops actually doubled from 3 to 6 countries, which are now cultivating the GM crops, and actually now is even more increasing. So the GM crops has you need the enabling environment, in order to see the full potential of these crops. But it might help if there is an enabling environment in the country, then it might help Africa tackle food insecurity and malnutrition, and as well as hunger. There is prediction that by 2025, actually the GM market in Africa will increase to about $871 million US dollar. Which is something good. Now moving to the newer technology, which is genome editing. So genome editing has become an important molecular tool for crop improvement. When we talk about genome editing, is not only one technology, is a group of technologies starting from meganucleases, zinc finger nucleases, talents, and then more later is the CRISPR-Cas9. So this technology is very precise and efficient, and it makes very small targeted changes in the cell of an organism. So basically, these endonucleases, they make the double-stranded cut, and then once this cut is done, actually is the machinery of the organism itself which makes the repairing. And then based on this repairing, actually there are either small insertions or deletions, or sometimes it can be replacement, or it can be like a whole allele or the gene replacement as well. Actually, genome editing, when we talk about that, is not a new technology. Nature has been editing, and that's how the whole domestication happens. So, nature edits, and then the farmers goes and select, and so it has been doing it. But those ones were more like a random mutations. So mutations has been accelerated in 20th century using chemicals and radiations. And later on, the technologies were developed for more precise gene-targeted mutations. For example, meganucleases, zinc finger nucleases, and talents. But in more recently, actually CRISPR has been, like in 2013, the CRISPR was first reported in the crops, and it has become now the most popular editing approach. And for that reason, CRISPR technology actually got the Nobel Prize in 2020. Jennifer Doudna and Carpentier Emmanuel, they got the Nobel Prize. So this technology has a lot of potential benefits, starting from the functional genomics, like you can use it for gene discovery, can be used for nutritional enhancement, yield improvement, resistance for the biotic and the abiotic stresses, as well as for the herbicide tolerance. This technology has been so far applied in more than 40 crops across 25 countries, addressing most of these traits. Actually, the crops might be even more, because this is the technology where I see much more publication coming out every week, you know. And so among all the different technologies used for the genome editing, actually CRISPR is about 60%, which is now used. And this is basically because this technology is easy to adapt. It is very, very precise, but also very easily you can also multiplex the trait using the CRISPR technology. So this is reality, because there are now the products in the market. There is a high oleic soybean oil, which is the first gene edited food crop on the US market. Japan has approved this GABA rich tomato, and these tomato actually can fight the high blood pressure. And recently, actually, in Japan, there is another which is also approved for the commercialization, is the WACCI corn. In addition to that, there is a non-browning mushroom which was approved in US and Canada. There is also this blight-resistant rice, which was approved by USDA and then by Colombian regulator as non-GM. But this is still not in the market. And then recently, actually, Argentina, Brazil, and Colombia approved gene-edited drought-tolerant soybean for the trials. In Africa also, in Kenya, actually, Kenya approved MLN maize for the field trial as non-GM last year, and recently actually they also approved the Striga tolerant sorghum as non-GM for the trials. So it has potential value because as I mentioned, it is relatively accessible, affordable, and then the public and the private sector can avail it. Another good thing about this technology, when it comes to the consumer's perception, actually it's much more positive for genome editing in comparison to the GMOs, because consumers feel it is more natural in comparison to the GMOs, which has always the foreign gene integration in them. It can actually, this technology can have implications on, like, you know, positive on the SDG 2, which is for the zero hunger, and also for the climate action. But in order to, again, to utilize these technologies, we need the enabling environment with the science-based regulatory guidelines for the release and the adoption of the gene-edited product. So I think in order to understand the regulatory implication about this technology, there is need to understand what type of edits the product will have. So as I mentioned before, you know, these the site-directed nucleases, actually they make the double-stranded cut, and after that the repair happens. So when the repair happens, the repair can happen two ways. One is this on non-homologous end joining, and in this case, actually the cell's own machinery actually do this repair, and during that repair, actually they can have some targeted mutations. So most of the time it leads to the gene silencing or the gene knockout, and it is called SDN1. The second way of repair is when there is a donor template present, and then this is the homologous directed repair, and then it can be like either a small insertion, and so this is SDN2, and then this is also very targeted, and then the third type is the SDN3, where it can be the whole gene insertion or the allele replacement. So SDN1 and SDN2, these are the type of the mutations which are very similar to the conventional mutations which can happen either through the chemical mutagenesis or irradiation or even the spontaneous natural mutation. But SDN3 type, the regulation depends upon the integration of this. So this insertion, it can be the cisgenic if it is coming from the same plant species, or it can be transgenic if it is coming from the outside of that plant species. And then the regulation of SDN3 also depends upon the country regulation. Some of the countries actually regulate them as GM regardless of whatever the insertion is from, but several countries, depending upon if it is cisgenic, they are not regulated as GMO. So this is currently the global legislation of genome editing. Actually, this graph is another one which is actually changing very frequently. So the countries which are in green, shaded in green, these are the countries actually where the genome editing guidelines is already in place, and based on that guideline, if there is no foreign gene integration in them, they are not regulated as GMO. The countries, so in Africa, actually there are 3 countries, Nigeria, Kenya, and Malawi, where the guidelines are already in place. So I think one point I would like to mention that when it comes to the genome editing, actually the regulators are actually using very different approach what they used to use before. So for Kenya and Nigeria, both they have done lot of homework before putting this guideline in place. So they have actually reviewed the guideline from different countries like US, Canada, also Europe, some part of Asia, but also from Argentina. And Argentina is the first country who put the genome editing guideline in place in 2015, but also is one of the best guideline, I will say. And Kenya has actually adopted the guideline from Argentina, which is like really commendable. And then the countries which are shaded in yellow, these are the countries where the guidelines are actually under discussion. And like Ethiopia, Ghana, these are the countries which are like really in very advanced stages in the approval of the guideline. And UK has another good example which has actually approved. Initially they approved it for the field trial, but recently a month back they have also approved the guideline where the gene edited products, if there is no foreign gene, they are not treated as GMO. And the countries which are shaded in red like South Africa, Europe, and New Zealand, these are the countries where actually they treat gene edited and GMO similar, but still there is a challenge because not all countries has the similar guideline. The guidelines are really different because only one common thing is the SDN1 is in all these green shaded countries are actually treated as non-GM. So if we talk about the different improved crop varieties, this is the scenario, I'll say, in terms of the biosafety regulation, because none of the new varieties is non-regulated. You have to go one way or either, some sort of regulation. But now what I'm talking about is the biosafety regulation. So if they say there are the new varieties coming out from the breeding program as the conventional varieties, they are not regulated. When you have the genome editing, actually the SDN1 type, if there is no foreign gene integration, they are not regulated as GMO. SDN2 type also in most of the countries are not regulated, apart from some countries which has very clear, like only SDN1 are not regulated. And SDN3 type, as I mentioned, it depends upon what's the insertion, whether it's from the same plant species or from different plant species. And definitely the GMOs are biosafety regulation there as well. Okay, so once you have the genome edited products, then the first question is whether there is any foreign gene integration in them or not. If there is a foreign gene integration, then they are treated as GMO, and you follow the guideline specific to your country for the commercialization of the product. So that means you have to do the risk assessment and the biosafety, or all the biosafety requirements for the country. But if the products are non-GM, they can actually follow the same path where the conventional varieties are following, and in the end you go for the seed registration and the release. So this figure shows what's going on already in Africa. It might not capture everything, but at least it's capturing most of the things what's going on. So, for example, in Egypt, they are working on the drought-tolerant wheat. Ethiopia is working on teff for semi-dwarf logging resistant. And they are also working on improving the oil quality of the Ethiopian mustard. In Uganda, they are working on cassava. And you will hear more from John about that. In Kenya, actually there is a lot of projects going on. There are projects on the disease-resistant banana, also on yam. There is MLN-resistant maize, striga-tolerant sorghum, virus-resistant potato. These are the crops, but there are also a few projects in the livestock which is going on there. Even though the South Africa is red, there are still projects going on in South Africa. One of them is the virus-resistant cassava. In Nigeria, they are working also on cassava for the disease resistance, also the enhancement of the micronutrients. In Ghana, also there are projects going on sweet potato, and then maybe other crops. And in Burkina Faso, actually on rice. So what I'm going to do is I've talked about the gene editing and this also the biotechnological tools. Now I will give you an example, like, how we can apply. And because my lab works on banana, so the example I'll give you from banana. So as you know, banana, plantain, very important crops. I don't need to mention that, you all know that. But what you don't know is, like in banana and plantain, there is also the huge yield gap. So this is a cooking type of banana, which in East Africa, they call it matoke. You can see here, so this is the actual yield and this is the potential. And the similar thing is for the plantain. So there are several reasons of this yield gap, but one of the reason is actually the major reason is the diseases and pests. So banana is affected by a lot of diseases and pests, fungal diseases like black sigatoka, fusarium. There are bacterial disease, and there are also viral diseases like banana bunchy top and banana streak virus. And then there are pests like nematodes and weevils. The problem is not that the crop is affected by so many diseases and pests. The problem is this, that they are coexisting. So on the same field, you can find out there are a lot of pathogen and pest affecting the crop. As you can see in this graph, this multicolor spots are like different pests and pathogen. So at IITA, we have a very strong crop improvement program for banana. And what I'm now trying to tell you here, that is not only the biotechnological tools. We need to use all the tools available to us. So we start from the selection from the existing germplasm. Then we have the conventional breeding program. We also do a little bit of mutation breeding. And then we have transgenic and genome editing. And based on the need, we apply the technology. For example, we can do the conventional breeding, but conventional breeding in banana is comparatively, it takes long time, but it has low cost. But the major limitation of the conventional breeding is that if you don't have the resistance to this disease in the germplasm, then you cannot cross. So if that's the case, then we move to the biotechnological tools. For the transgenic, we bring the genes from outside of the banana species, so that is a foreign gene. And for the genome editing, we are actually trying to tweak the genome of the banana itself to make the resistance. So currently we are working on 3 different traits for genome editing: the banana streak virus, the banana xanthomonas wilt, and the fusarium wilt. So this example, you will find it very interesting because the banana streak virus is a double-stranded DNA BATNA virus. And this virus actually integrates into the B genome of the banana. So basically, banana comes from 2 diploid progenitors, AA, which is one parent, Musa acuminata, and BB, Musa balbisiana. And then most of the cultivated varieties are either triploids, so either AAA or AAB or ABB. But there are some tetraploids as well, some of the hybrids which are released. So this virus is only the problem when the banana has the B genome. So particularly plantains, because plantains are AAB. So this virus is a big problem in the plantain. So banana streak virus has a monopartite genome, like here. It has 3 open reading frames, open reading frame 1 and 2, which are relatively smaller, and they are involved in the virion assembly. And then the open reading frame 3, this which you see here in red, is the big open reading frame, and it basically encodes all the essential genes required for the viral replication. And so this open reading frame, it actually is a polyprotein, and which gets cleaved at the aspartic protease and in order to make these functional proteins. And when this virus is integrated into the banana genome, it normally gets integrated into as a multiple tandem repeats, direct or indirect tandem repeats here, like you can see here. And when this is integrated, it sits very quietly, and that's why it's also known as endogenous, because it has become part of the banana genome, and plant doesn't show any symptom at all. But under the stress condition, so this stress can be environmental stress, like drought, or increase in temperature, or even the tissue culture, because banana is vegetatively propagated and is multiplied using tissue culture. So tissue culture is stress, and also during crossing, banana plant feels stress, and under that stress, this integrated viral genome gets activated into this episomal form, and that's when they showed the symptoms like this, the very beautiful streak. This virus is actually a millibug vector which is spread, but most of the time is not because of the millibug, but because of the activation of this. That's when the endemic happens of this virus. And so this has become, particularly the plantain has become like a major challenge in breeding, as well as the dissemination of the hybrids, because many countries, they don't allow plantains movement because of this virus. So germplasm movement is a problem because of this virus. And also breeder don't use the diploid Musa balbisiana, which has the BB genome, and even though this diploid has lot of resistance against several diseases, but they don't use it into their breeding program. So the strategy which we used was actually, if we can somehow make this virus inactivate so that it doesn't activate into the episomal form, that was our strategy. So what we did was, we designed the guide RNA to create the mutation in all the 3 open reading frames. And in open reading frame 1, 2, but then in the 3rd open reading frame, I actually decided to create mutation in the aspartic protease, because my idea was that if I make aspartic protease non-functional, basically I'll make the whole open reading frame 3 as non-functional, because the cleavage will not happen of the polyprotein, and then the functional proteins will not be there as well. So what we did, we used a plantain, Gonja manchaya, which is a very commonly grown plantain in East and Central Africa. And so we took the plants which were non-symptomatic from the field, cultured them into the lab, and then after that we did the characterization to know the integration pattern. So we saw this integration after doing a lot of molecular characterization, and we realized that these ones has only the integrated version, not the episomal, and that's why the plants were not showing the symptoms. After that, we have cell suspension of this variety already in the lab, and we created 3 guide RNA targeting, as I mentioned, the 3 open reading frame, and we delivered these CRISPR-Cas reagents into this cell suspension and generated the complete plantlets. So these are the plantlets, like these are the edited and the non-edited, and then we did the target sequencing to check for the targeted mutation. So we saw a big deletion, about 198 base pair deletion of the viral sequence in the open reading frame 1 and 2, and then some indels in the open reading frame 3. Because we designed the guides very specific to the viral genome which was integrated, so that's why all the mutations were in the integrated viral genome. There was no mutation into the plant genome. We actually did quite a work to check for the off-target mutations, and we didn't find any off-target mutation in these plants as well. After that, we evaluated these plants in greenhouse. We actually mimicked the drought, so we didn't water these plants for some time, and so this is our control wild type, which shown very good symptom under the drought condition, and these were our mutants. So majority of our mutants didn't show any symptom apart from these two. And so then we actually correlated the sequencing result with this, and we find out that the asymptomatic mutants had the mutations in all the open reading frame, including the 3rd, but then the 2 which were showing the slight symptoms, actually they didn't have the mutation in the open reading frame 3, which is very much what our thinking was as well, that the mutation in aspartic protease is critical because it makes the whole open reading frame 3 non-functional, which actually encodes all the major genes required for the virus replication. And so if you need more detail, this is already published, so you can read it. And we also check the viral load, and we didn't find any active virus in this. So this one, the first example I showed was where we targeted the integrated viral genome, but not doing anything to the plant genome. Now the second example I will show is where we have actually done editing of the plant genome. So this is about the banana xanthomonas wilt, because this disease is one of the major devastating disease in East Africa. So the disease is actually, that's how you see it can wilt the complete plant, but the major problem of this is like the disease spreads very fast. Once it's in the field, actually it can wipe off the disease within a few months time, and because of that there are losses estimated from 40 to 80% depending upon the country. So in DR Congo actually is about 83% where the losses are, and the disease is actually in all the Great Lakes region of East and Central Africa, which is one of the major banana producing country. So we also did a lot of genotyping screening for the susceptibility of the disease, and so we found that all the cultivated varieties of banana are susceptible, but only Musa balbisiana found to be complete resistant to the disease. And then there are some tolerance in Musa acuminata subspecies Zebrina and Benkesai. But you remember I mentioned that Musa balbisiana cannot be used into the breeding program because of the problem of the banana streak virus. So we cannot use this in the breeding. and the estimated economic losses are about 2 to 8 billion. And this is the scenario. Once the disease is in the field, you have to uproot the plants and leave the field fallow before you can do the replantation as well. This was initially, long back when we started working on this disease, we actually went through the transgenic approach, because that time the CRISPR was not even there. So we used the transgenic approach, and what we did was we used 2 genes, PFLP, plant ferredoxin-like protein, and HRAP, HR assisting protein. These are the defense genes from Capsicum annum, and we cloned these genes, and then we have overexpressed these genes in bananas. So we developed the transgenic banana. We have a very rapid bioassay which we use to test. This is our control plant. We have artificially challenged these plants with the bacteria, and you see the control plant completely wilted, and then these were the plants which has not shown the symptom. So we did this assay for 60 days, and after 60 days, the plants which didn't show the symptom, we actually further progress those plants into the greenhouse screening. These are our control plants. You see they all got wilted after challenging them in the greenhouse, but then these were our transgenic events which didn't show any symptom at all. So we also tested, to try to isolate the bacteria from the 5 centimeter above and below the inoculation point, and we didn't see any bacteria there, because these are the hyper response assisting proteins, so they create the physical barrier so that the pathogen cannot move beyond that point. So that was the theory. And so these transgenic plants, in collaboration with NARO in Uganda, actually we tested up to 3 different generations. So that means 5 years in the confined field trials in Uganda, and they didn't show any symptoms. So these are our transgenic plants, and these are the control plants which were completely wilted. So these plants, these were the Uganda, the varieties which are farmer preferred varieties in Uganda, and because Uganda, so these plants are still in Uganda there. So right now what we did, you remember I mentioned that the Musa balbisiana has the complete resistance against this disease. So we did lot of molecular characterization, and now what we are doing is we are taking this information from the Musa balbisiana and we are transferring that information into the susceptible varieties, either knocking down of the susceptibility genes, or we are overexpressing the defense genes. But in this case, these are the Musa genes which we are trying to overexpress. So what we did in order to get the molecular basis of the disease resistance in Musa balbisiana, we did the comparative transcriptome analysis, where we did RNA-seq at the early stages of infection in the Musa balbisiana compared to the very susceptible banana variety called Pisangawak. And based on this, we got a lot of differentially expressed genes, which we have mapped them to the Biotech pathway, but also the metabolism pathway. And from there, based on that, we identified the targets for genome editing. So we find out a lot of targets. Some of them are the susceptibility genes, some of them are the transporter genes, or the transcription factors involved as the negative regulator. But we also identified a lot of defense genes which we can also overexpress. So we did that. So a lot of several Musa defense genes we have tested, like antimicrobial protein, some of the receptor kinases, as well as the pathogenesis related genes and the disease resistance genes. Initially, we tested them individually, and then based on the result, we have now actually stacked 3 together, because the disease resistance based on the single gene has chances for breaking down of resistance very quickly, because as the pathogens evolve very nicely as well. So we are now stacking, and now these plants are ready for going for the field trials, maybe early this by the end of this year. So on the susceptibility genes, we tested a lot of susceptibility genes, but the 3 of them actually showed us good result. This is the result from the pub 2223. We created the double mutant and we challenged them in the greenhouse, and we have the plants which has actually completely resistance to this disease. And then another example is the DMR6, which is very well characterized susceptibility gene. So we have done the DMR6 ortholog from banana. This susceptibility gene actually gets highly activated up to like 60-fold in banana in reaction with the interaction with the bacterial pathogen. So we have again designed the guide RNA against that, and we created the mutants, and we have screened these mutants initially in the lab and then in the glass house. And so these are our mutants which has not showing any symptom, but these are the control plants which showed completely wilting. And then we have the targeted mutations identified in that one. We also checked for if their effect on the plant growth, and we didn't find any negative effect of the plant, of knocking out of this gene on the plant growth as well. So one important thing is, like, because this is a new technology, we need like really the critical mass in Africa in order to, like, really utilize this technology. So what we are doing is we are engaged in a lot of capacity building, and the capacity building is at all the level, the researchers, but we are also in the early stages, we also involve a lot of regulators so that they understand the science behind that, and also the journalists. So these are the journalists who are visiting the lab, looking at the plants themselves, so that they can see how in the reality these plants look like. And then we are actually partnering with Alliance for Science with a lot of training for how to communicate the science behind genome editing. We are using different platforms for sharing the stories, and I'm training a lot of PhD students. So 3 PhD students has already been graduated training on the genome editing, and 3 are currently working in the lab. And another good thing which we'll say in a partnership we are doing is we are partnering with UC Davis and UC Berkeley, and we are training the scientists, the African scientists, for CRISPR technology under the African Plant Breeding Academy for CRISPR. And so this one our aim is to actually train about 80 African scientists within the 5-year period, and I'm very happy that we are working with Professor Rita Mom on that one, and other partners there. And so this year the first batch has started in January this year, and there are 11 students from 7 different countries, and we do have the student from Ghana as well. So the key take-home messages are, in order to feed all the people, the increasing population, we need to at least double the agriculture, and Africa could produce 2 to 3 times more of these staple food crops. They have the potential for doing that, but in order to do so, we need the agriculture innovation. And CRISPR technology can be one of the very powerful tools which we can utilize, and it is very simple and efficient. You can use multiplexing, and it's easy to adapt. Genome-edited crops with no foreign gene integration are treated similar to the conventional crop in several countries. The application of CRISPR-Cas-based editing has been demonstrated in several crops, about 40 or maybe more now, and I have shown you example how we can apply for the disease resistance in banana as well. So in the end, I would like to acknowledge my team from IITA, our different partners, and also the financial support from USAID and the CGIAR research program. Thank you very much, and I'll be happy to take any question.
Thank you very much, Dr. Lina. We do not have a lot of time for now, so we're going to take only 3 questions, and then we'll come back from the break. If we come back in good time, we'll have 3 more questions. Good deal? Bon, mon frère, on commence avec toi. Oui, please, I'll come to you. You are number 2.
Thank you very much.
And please make it very crisp, straight to the point.
Thank you. Please, I need a clarification. Musa balbisiana, you said, yeah, breeders don't use it. Is it because of the susceptibility to disease or because of some crossing barrier? Because you mentioned that it is tolerant in one place, while the Musa acuminata, something like that, is also resistant. So, a clarification for that. The second question is that based on your experience, or maybe the possible consequences, what are the possible consequences of overexpressions? When the gene is overexpressed, what are the possible consequences or your experience in terms of the phenotypic expression or yield or test? Is there any things like that that is affecting those? Thank you.
So those are 2 questions. We'll take one more. Please, let's have yours.
So fantastic talk, very impressive. So the banana work with the gene editing, I mean, it's really impressive that you can take out this intrinsic virus, but presumably you still have the transgene there, right? And we talked about this earlier, but mechanisms to actually have it as a truly gene-edited crop and not as a gene-edited crop.
Yeah. So I will start with the last question first. So basically, for the plantain work, for the streak virus, our approach is different. So we got the proof of concept where we use the plantain, the triploid, but now we are going back to the parents. So there is so that only one cross needs to be made. So it's like a diploid, the improved diploid, and the tetraploid. So we are trying to actually add those ones so that those can be crossed, and then our final product will be non-GM because we will segregate out. So that's what our approach is for the plantain. But for the triploid, AAA, that's difficult. So we are trying different approaches like protoplast, also the excision, what we discussed before. So we are trying different approaches there. Related to your question, actually the Musa balbisiana has, as I mentioned, has resistant to bacterial wilt. And then it has also resistant to few other diseases. But breeders don't use it. One of the reason is that the streak virus is integrated, like it has the B genome. But yes, you are right. There are other attributes also of that wild type progenitor, and for that reason also breeders don't use.
And I look forward to the communique on pathways to making Africa food sufficient, which will be issued at the end of this symposium. We at the Minister of Food and Agriculture will be looking forward to the recommendations you will make in that communique for ideas on what we can also do differently to help transform Africa's agriculture. Congratulations to the West Africa Center for Crops Innovation, WACCI, and your founding director, Professor Dankwa, for your role in enhancing food security over the last 16 years, not only in our country, Ghana, but the rest of Africa. Happy 16th anniversary to you all, and congratulations to the team at Alliance for Science Ghana, led by my good friend, Joseph Opoku-Gakpo, for putting together this magnificent symposium. I wish you all the best in the deliberations at the symposium today. Thank you for your attention.
So please, kindly exit now. There are ushers outside who will lead you to where we would have the group photograph. Joseph, that is sorted? Yes, so we'll take the group photograph and then we'll return, grab a coffee, and come back here at 11:15. Thank you very much. See you in a bit.
2. 1, 2.
They used the events today. Mother, when you own bit.
you. Africa at the West Africa Center for Crop Improvement. I'm here inviting you to this exciting program on biotech crops. It's an international symposium on biotech crops, and I'll be speaking on a panel talking about the future of biotechnology crops in Africa. You are all invited. If you cannot join us in person, please try and make it online. Join our social media.
Let's come in. Those of us who are still outside, let's come in.
Of June 2023.
All right, welcome back, distinguished ladies and gentlemen. Please, multimedia, take down the videos. We are about to begin the second half of today's program. We'll be having discussions from Joseph Opoku Gakpo, Dr. Jerry Mboyini, Dr. Nigel Taylor, Dr. Murenga Mwimali, Dr. Edgar Traore. So I'll be inviting them at the right time to start. So ladies and gentlemen, we begin this second session with an overview of Africa's GMO landscape. We'll be examining which countries are growing what and what best-in-class lessons there are to learn from them. The first to take us through this, and he has exactly 20 minutes to do this, but we appreciate if he does it in 11 minutes or 12, will be a research fellow at the Genetic Engineering and Society Center, North Carolina State University, USA. He's the main speaker for this session, Mr. Joseph Opoku Gakpo. Please, let's welcome him with a round of applause. In the meantime, we are streaming live on our social media platforms, and if you find any comments interesting or you've taken any photos you want to share on social media, please feel free to do so, and hashtag it, hashtag Future of Biotech, and also tag Science Ally. So please feel free to do so, hashtag Future of Biotech, and also tag Science Ally. Science Ally, one word. All right, thank you very much. Joseph, please.
Thank you very much, Chris. I won't even take the 20 minutes that you indicated I have for two reasons, because a lot of what I was hoping to say during this session has been said by Dr. Tripathi already. And I think those who are working on the specific crops, the specific GMO crops, we need to hear from them rather than those of us who are running commentary about what's going on. So I will quickly run through this. The final bit of that is where my interest is. I'm a fellow at the Genetic Engineering and Society Center, and myself, what can make at the center, and then most importantly, the center focuses on the technical, the ethical, and the societal dimensions of products and the impacts of biotechnology. We'll come back to that a little later when I'm rounding up this session, because then the idea of interdisciplinary research when it comes to work around biotechnology is an important thing that we are hoping that we would all be paying attention to going forward. A quick overview of GMOs in Africa. I picked a lot of the data here from the Genetic Literacy Project. The indication is that a lot of the crops that are being developed using GMO technology across Africa is actually being done by public sector scientists, which some say is an exciting thing because then you are doing it for the public good. But then there is also the concern that also raises issues about continuous funding and whether you would have the necessary funding to do that, as against when it's private sector funded, when there is almost a guarantee that the money would come in. About 8 countries have commercialized 4 different GM crops currently in Africa: cotton, cowpea, maize, and soybean. And even as the Deputy Minister indicated, out of the 54 countries we have in Africa, only about 15% of them have various GM crops. These are the specifics in terms of the various crops that some of the countries are doing. I won't go over it. The slide will be available for us all to see. There are 2 countries that they approved GM cotton for use, but then the approval was subsequently suspended. The Burkina Faso example is an interesting one. Dr. Traore will be speaking about it soon, so I won't touch on that much. But then some of the traits that there's been a lot of focus on include drought tolerance, nitrogen use efficiency, salinity, and crop composition, as well as disease resistance. And this specifically has to do with crops which are still currently under development, even away from the earlier ones which were approved, which were mainly focused on herbicide tolerance and then insect resistance. But these are some of the traits that are being worked on in various confined field trials. There are about 33 of them that are ongoing across different African nations. This is something that we pulled from Bruce and Barefoot's 2018, which then tells the story of the amount of GM crops that are being grown on the African continent. So we see the USA all over the world, they are accounting for about 36% of all the GM crops that are produced. Brazil comes next, somewhere around 30%. Africa lies in the others angle, below 7%, and the indication is that Africa actually accounts for somewhere in the region of 1.5 to 2% of all the GM crops that are grown worldwide. This is also data on the kind of earnings that we've seen over the years from GM crops, the estimation being that when it comes to the additional benefit that GM crops have brought, 261.3 billion is the total amount that has accrued since 1996 up until the year 2020. Africa's earning within that space is also within the other countries' bracket, just about 5.7% of that amount. This is despite the reality that a lot of data has actually reported the benefits of GMOs generally, including how when it comes to comparing them to conventional crops, their GMO counterparts have created situations where there is an average of 22% yield increase for farmers. There is about 68% increase in their profits. The technology has played a role in reducing carbon emissions into the atmosphere. And despite that evidence, we all know that the penetration of GMOs across Africa has been a very difficult thing. Some 3 specific case studies that we sought to draw attention to. South Africa, first of all, they are kind of the leading country when it comes to the growing of GMO crops on the African continent. They had a biosafety law passed in 1996. Well, they jumped on the technology soon after the commercialization, even in the USA. They are currently the 9th largest producer of GM crops. More than 90% of their cotton, soy, and maize are GMOs. Burkina Faso in 2006 then also passed the biosafety law. They introduced GM cotton. More than 80% of their cotton was GM. Subsequently, due to a number of challenges that I know Dr. Gatraro will be telling us about, that process was suspended. And then Uganda is also the other interesting story, where pre-2017, there were a lot of GM trials ongoing. They attempted passing a legislation in 2017, which is still in limbo up until date because Parliament and the executive don't exactly agree on the direction that they should go. We are drawing attention to this specific case study to then point out one thing, that there is a benefit in being an early adopter when it comes to GMOs to a certain extent. South Africa went ahead and did it even before the growth of a lot of anti-GMO campaigns, and they've now become the leader. A number of the others who did it subsequently, including Burkina Faso in 2016, and Uganda, which is now trying to then get the necessary legislation in place, have struggled because of everything else that has actually happened. There's a point there that there's probably a lesson that we should all take based on what's happening when it comes to the use of the CRISPR technology that we heard Dr. Lina Tripathi talk about today. There is probably the chance that the later countries are in adopting, the possibly higher the chance that they'll struggle to then get it through, because then by then maybe anti-gene editing sentiments may have grown. So what has slowed the adoption of GM crops across Africa over the years. We identified some specific points, including concerns about inadequate human capacity. That's a source from 2011, but we know that a lot of changes have been seen in that regard over the last 10 years or so. WACCI is training a lot of people who have the expertise to be able to work on some of these crops over the years. There's the talk about the cumbersome regulatory procedures, which is something that we all know about. Lack of investments, which is one of the key things. In some countries, projects on GMOs have been suspended because the donor partners have pulled up funding and the various governments are unwilling to then continue those investments. There's also the talk about a lot of the focus being on farmer benefiting traits like insect resistance, herbicide tolerance, rather than focusing more on the nutritional bits which go directly to the consumer, also possibly being one of the things which have slowed the process when it comes to the spread of the adoption of GMOs. And then public misinformation, which is one of the things that Alliance for Science, where I'm a fellow as well, we pay a bit of attention to. So we did a study in Kenya early this year. It got published where we looked at misinformation when it comes to publications in the media concerning GMOs. And we found that 40% of articles published in the Kenyan media actually contained GMO misinformation, which went unchallenged. Only about 3% of those articles actually contained pro-GMO information. These very high rates of misinformation certainly plays a role in situations where the public gets misinformed and then it gets them to then have a certain hesitancy towards accepting some of these crops. Quick points on some of the lessons in terms of what possibly attention has to be paid to. Improving upon the human capacity and improving the technical know-how here in Africa when it comes to some of these crops. WACCI is doing a bit of that work by training a lot of PhD plant breeders. The concern about investment in research and development in Africa, which we drew attention to a little earlier as well, that's also one of the things that attention has to be paid to. There are concerns about political interference in the various regulatory processes, which we need to then avoid going forward. Human attitude towards new technologies, that's also come up over and over again. The point being that over the last 25 years or so, we should have learned the lessons about how Africans generally pay attention to some of these new technologies and respond appropriately to then ensure enhanced adoption. And then the concern about the cumbersome regulatory procedure, and then the issues having to do with the communication, where we are drawing attention to misinformation that attention needs to be paid to so that then adequate and appropriate information is rather sent to the public in Africa on GMOs. Quick recommendations. We're asking that there should possibly be interdisciplinary approaches to introducing GM crops that consider the social, the cultural, the economic, and other justifications for some of these technologies. All too often, then we are just developing the crops and we are paying attention to the science itself. That's why I was drawing attention earlier to what we do at the Genetic Engineering and Society Center, where we are interrogating the ethical bits, the technical, even beyond the technical bits, we are interrogating the ethical bits, we are interrogating the social implications. We say more attention is to be paid to that so that then the introduction of some of these crops are done in more holistic manners, using expertise from other fields beyond plant breeding itself. We make the point that GM projects should probably have economists, psychologists, anthropologists, so that then it's an all-encompassing approach rather than just a plant breeding project. And then, well, finally, we're recommending what we have at the GES, the style of interdisciplinary approaches when it comes to conversations around GMOs. That's one of the ways that our scientists in Africa should work to help enhance access to some of these crops. So thank you very much.
Thank you, Joseph. So we're going to take questions after all the presenters have spoken. So please keep noting down your questions. Dr. Jerry Mboyini is the principal investigator at the BT Cowpea project. He will be speaking to us about Ghana's first GM crop, the BT Cowpea. Dr. Mboyini, please.
Good morning, everyone. So I'll talk to you about Ghana's first GM crop. The good news here is that Dr. Batiano, Dr. Lawan, and then Dr. Ijoma are here, and they are part of this particular exercise. Then my GM police, Dr. Jamin, is also here, so at least I have a number of lawyers to bail me out. So I'm just a mouthpiece for the team. Unfortunately, most of them are here. So Ghana's first GM crop. Ghana's first GM crop, a number of people on the research team, and then, yeah, so I'll give you a brief introduction. In Ghana, we consume a lot of cowpea, and the common name over here is that we call it beans. Now, out of the 169,000 tons consumed annually, it is only 57,000 tons that is produced domestically. And the main reason is that we have a lot of pest problems on it, which result in low yields of the crop. Now, how do we cater for the deficit in the demand for the crop? The bulk of it is imported. So from Burkina, from Niger, and then from Nigeria. That's where we import from to actually augment the demand. We have the flowering stage, we have trips, and then we have the maruca or the legume pod borer. These are basically insects that feed inside the flowers. And because of that, when a farmer applies insecticide, it does not effectively control it. Then when the flowers survive and you have the pods coming up, all these pests and more attack the pods. Now you realize that at the flowering stage, I talked of maruca. Now at the podding stage, maruca comes there again. So it is a pest at the flowering and also at the podding stage of growth of the crop. And these are the main reasons why we have very low yields of the crop on farmers' field. So what does maruca do? They will feed on the young leaves. Then they will damage the flower bars. Then they will feed on the flowers. And also, and basically all these things are done inside the flowers. So insecticide application is really not effective on it. Because of that, farmers typically record up to 80% yield loss if spray applications are not very successful. Currently, our farmers, we don't have any resistant variety for maruca. So we have varieties of cowpea that are resistant to striga, varieties that are resistant to aphids, but we don't have some for maruca because conventional breeding has not been very successful with that. Farmers typically spray 8 times, and then the harmful effects on the environment, we are all aware of it. So basically, Ghana joined the pod borer resistant cowpea project somewhere in 2012. It is a multi-country project, and the basic aim of that particular project is to ensure that the livelihood of farmers in sub-Saharan Africa can be improved by delivering better cowpea varieties that are able to overcome damage by this particular insect known as the legume pod borer or Maruca vitrata. We went for the genetic modification approach. Why? I mentioned it earlier. After screening over 15,000 cowpea accessions, there was no source of resistance identified that could be used for conventional breeding approaches. Because of that, the only option was to rely on the use of the BT gene that has been proven worldwide to be very effective against Maruca. So the CRY1AB gene was the first one that we worked on, and it's from Bacillus thuringiensis. The genetic modification was basically using the agrobacterial-mediated transformation, and then we had the MPT2 protein as the selectable marker for it. So it encodes for the CRY1AB gene. I mentioned earlier that this is not just a CSIR SARI activity, but then it is a multi-country project. Our work is coordinated by the African Agricultural Technology Foundation. The headquarters is in Nairobi, but then for cowpea, it is headquartered in Abuja. Dr. Ijoma is here, she's from that particular place, and a proud product of WACCI. So for the participating institution, we have the CSIRO in Australia, then SARI, and then INERA in Burkina Faso, then IAR in Nigeria. What I didn't add was that we have the Danforth Institute also partnering us in this particular work, and we are funded by USAID. So this is a front view of our confined field trial site at Nyankpala when the whole work started. somewhere in 2012. So it's a restricted area with a security post to ensure that nobody trespasses without authorization. I like this particular photo because the first principal investigator for the project was Dr. Atokple. He was the first principal investigator for the project, and then that was him planting the first BT cowpea in Ghana, and that was in September 2013. Then that particular trial, we started off trying to test our materials that had a BT gene in it for resistance to the legume pod borer, so that those ones can be used as donors for introgressing the gene into promising commercial cowpea varieties that are released in Ghana. So we started off, so this particular IITA line was the first one to be transformed, and then out of that transformation, we had about 6 promising events that came out. So we started by looking at this transformed event and comparing to select a candidate that will be used as a donor. And basically, that work involved putting about 40 larvae of laboratory-reared maruca on flowers and pods of cowpea at bi-weekly interval, just a high infestation pressure beyond what happens in nature, so that we can really be sure that we have any candidate that we select as a donor is really effective in controlling the pest. So from those works, we selected event 709A as the most promising candidate. And over there, you can see Dr. Atokole performing crosses to actually introgress the gene from the event 709A into a release variety, which was known as Songotra. Because it was a multi-country project, we selected Songotra because the same material had been released in Nigeria and Burkina Faso. So for the purpose of having a unified data set, we selected that particular line. Now, once we got the gene introgressed into Songotra, we moved ahead to do a number of tests on the Songotra without the gene, and it gave us excellent results. Generally, Songotra has a yield potential of about 2 tons, and we were able to hit that particular yield potential compared to non-transgenic, where we got less than 1 ton per hectare. So we did other agronomic trials to actually demonstrate that the genetic modification didn't have any unintended effect. Safety of the protein, that's the CRY1AB and the MPT proteins. We did compositional analysis, we did environmental risk analysis for weediness, polymediated gene. We had all these assessments conducted. Then we also needed to determine, so apart from maruca, other pests are on it, so how many times do farmers have to spray? After doing that, we realized that farmers needed a maximum of 2 insecticide sprays to actually protect the cowpea from other insect pests that were there, and that was a great saving to the environment. We worked with a lot of farmers, and basically they were brought to the confined field trial sites, which we have in Nyankpala, Damango, and Manga, and then they were to test the materials compared to their varieties and tell us which ones they preferred. From all the ways that we did, farmers always selected the BT cowpea. We had a number of engagement with farmers to actually introduce the material to them through field days in Nyankpala, in Manga, and all the other locations. Once we were convinced, we had the Institutional Biosafety Committee meeting to actually review our application that we were to submit to the Biosafety Authority. And this picture is from one of their meetings. When the Institute gave us the go-ahead, we were at the Office of the National Biosafety Authority to actually apply for environmental release. And the Honorable Deputy Minister told us that approval was granted in June 2022. That was last year. And this is the Biosafety Authority. That's the chairperson for the Technical Advisory Committee, actually handing over the decision document to our IBC chairman at SARI to show that they have approved for environmental release of the first event, which was 709E, and that expresses the CRY1AB gene. So once we had that, we conducted multi-locational trials last year. Then we also had participatory varietal selection also coming on. We are yet to do sensory analysis, and we intend to actually attempt applying for varietal release this year. For the multi-locational trial, we worked in 15 different locations in all the 5 regions in northern Ghana, and we worked with over 100 farmers from all the 15, from all the regions. Now, this is how far we distributed the sites where all these trials were actually conducted. And these are just a few shots of how the fields looked like last year when we went around for monitoring. The Biosafety Authority is yeah, you don't need statistics to be convinced that the transgenic is outperforming all the conventional materials that we actually tested, and this for green yield. Then these are from the farmers. So these are works that were done. These are results from the farmers for average for each of the regions. I presented for 4, though we did for 5 regions. And even on farmers' food, farmers are recording close to 1.5 tons, and this has never been recorded in cowpea for in all our conventional cowpea plot. So basically, what is the take-home message? What I've been trying to say is that yes, Ghana is on the path of releasing its first genetically modified cowpea variety, and all things being equal, we hope to make a lot of progress this year, 2023. This is not just something that came overnight, but it started since 2013. So it's a decade-long work with a lot of obeying restrictions from the regulators and then adhering to protocols that will ensure that we ensure safety. There's been a lot of diligence, and we've been meticulous in our data collection just to ensure safety and efficacy of the gene, because it is in our interest as public sector researchers to ensure that the right things are actually done. Generally, among farmers, there has been a unanimous acceptance of the variety, and this mainly because of increased yield and then reduced insecticide yield. The area of negative toll of pesticide use on human health, as far as cowpea production, will actually be a thing of the past once we have this particular variety out, because farmers are eager for it. Pesticide-related poisoning will definitely come down once this is adopted. And I must say that from a work that was done by IFPRI, we are actually predicted to benefit by over $79 million just from adopting this particular technology. And the delays that we've experienced in the last 5 years actually cost the country over $24 million. This was a survey that was conducted by IFPRI. Just to acknowledge that our work, like I said, we are coordinated by AATF. We have the Danforth Center, CSIRO, also being part of the partnership. Thank you very much.
Interesting how you committed your professor in the middle of your presentation. Very well. So we have 2 more presentations on this before we take the questions, and I'll kindly indulge your adherence to the timing we'll be giving you. So 10 minutes each. Dr. Nigel Taylor will be next. He's the principal investigator, virus-resistant cassava for Africa, deployment project, Donald Danforth Plant Science Center, USA. 10 minutes, sir.
Okay, thank you. I will do my best for 10 minutes. You gave me 15. Yeah, I'm going to talk about the WACCI project. I was here, I think, in 2018. I'm looking for Eric now. And I described the project then. We've moved on quite a lot from there, and we're getting tantalizingly close to delivering this product to the farmers. So I'm going to tell you about that. I'm going to skip over this. I'll skip over a few slides because of the time constraints. How do I find, where is this button here? Our goal is to develop and deliver cassava variety genetically enhanced for resistance to virus diseases. And I'll say this at the beginning because people will ask me. No IP restrictions for the end users and no charge by the technology developers. We are engaging with the private sector, so we are expecting people to pay, farmers to pay for the product at the end. And I'll describe that to you as I go along. Sorry, learning how to use this button, this thing here. 2 major diseases suppress cassava yields. Cassava mosaic disease caused by Gemini viruses and cassava brown streak disease caused by 2 epomoviruses. I'm going to focus on brown streak disease and tell you about that. Both of these are transmitted by whiteflies and, of course, by farmers that plant infected cuttings to establish the next cycle. Estimated to cause about $1 billion worth of losses to African farmers in a year. So cassava brown streak disease started off in East Africa, it's moved into Central Africa, and is coming to West Africa. So you may not think about it too much here every day, but it is predicted it's going to get here. And this is what happens to a Nigerian variety called TMS 0505 fine face. This plant dies when it's infected with brown streak. So it's not just the loss of yield that we see here, where you cannot eat this, you cannot sell it, and you cannot give it to your, you cannot feed it to your animals. It's actually plant death in some cases. So in our project, we're working with the National Agricultural Research Systems in Kenya, Uganda, Rwanda, and Nigeria. We have communication professionals involved, the ISA, and we're working now with Meta for the seed systems and delivery components. So we're a multinational, multidisciplinary team, about 50 people. I'm not going to read all of these. You can do that yourself. And these are the different areas that we focus on. All of this is needed if you're going to develop and deploy a biotech product. So one of the things I remind people is when we came along and our idea here was we're going to make cassava resistant to cassava brown streak disease. Actually, this is not conceptually a new idea. This has been achieved in papaya, plum, beans, squash, and other crops. And we do that by taking a piece of the viral genome, usually the co-protein, integrate that into the plant, and that then primes the plant, fires up its defense mechanisms, so it will recognize the incoming pathogen and be resistant to it. I'm going to go through this really quickly. We made the transgenic plants at the Danforth Plant Science Center way back early in the project. So did I do something wrong? There we go. And when we put these into the greenhouse and challenge them with brown streak disease by grafting, this is what happens. We get little storage roots about the size of your thumb, and you slice them open, this is what you get. Infection, disease, these are fully infected. Our transgenics, this is what they look like, at least some of them. No symptoms whatsoever, and when you do PCR on this, you cannot detect the virus. So we were really enthusiastic about this, and that meant get to the field. We went to the field, we did these trials in Kenya and Uganda. Namalonge, if those of you who know, Namalonge is a hot spot for just about everything, especially brown streak disease. And in this case, we were getting close to a 20-fold increase from the original susceptible variety just by expressing a little piece of the co-protein. So that was very, very exciting proof of concept for us. So we then really moved into the product development process, starting what I just described was between about 2009 and 14. And those of you who have been through a product development process, you will recognize this, all the different steps that you needed to do. And this is what we were doing, confined field trials, multi-locational field trials. We entered into a breeding program once we had identified our lead event, and also at the same time regulatory. You've got to go through regulatory. This is a GM product. And I will tell you briefly about that regulatory approval and where we are today in national performance trials. Still struggling with this, I apologize. I'm going to fly through this. The regulatory, if anybody's really interested in regulatory, please come and talk to me. It's very, very important. One of the main things that we did here, which was novel, we had regulatory field trials in Kenya and Uganda, and we combined the data. We talked with the regulators first, we combined the data to make one application. So this was a really good case of working together, data transportability, and engaging with the regulators early in the, throughout the process. We submitted our regulatory application to the National Biosafety Authority in Kenya in March 2020. We went through, all through this process, engaging with the stakeholders. This was a public forum in 2020, right in the middle of COVID, which of course didn't help us. And we had over 5,000 people attend. We engaged over 5,000 people in this process, stakeholders right through to the public, and we got our approval in June 2021. that Event 4046, one of our RNAi lines, one of our best ones, and progeny thereof, which is really important, as safe food for feed in the environment and allowing us to go forward to national performance trials. What we were doing at the same time is we were taking our lead event 4046 and a backup, and we were crossing it through a breeding program with breeders in Kenya and Uganda with local varieties. Some of you will recognize these, which are resistant to CMD and carry other beneficial traits. So we produced a cassava breeding program is you produce a large number of seeds and then you screen the F1s to pick out the best. And that is what we did through this. I'm talking to breeders, so I don't need to go into much detail here. Single plant where you collect a little bit of data, you bulk that up, you collect more and more data. So this is not just about disease resistance to CMD and brown streak. We're now looking at all the other farmer and consumer traits, right through to multi-locational trials across 6 locations in Kenya and Uganda, more than 50 combined field trials to date to get to this stage. So this is where we are. This was roots harvested at Mombasa about 3 months ago. This is our original TME204, non-modified. This is one of our F1 lines now, event 4046. So this has the transgene integrated into it. It has been in the field for 5 years consecutively. It has no symptoms, and we cannot detect virus in these varieties. The yields are very good, up to 80 tons per hectare fresh weight on Mombasa. If you've been to Mombasa, you can grow massive cassava there. And in Uganda, 35 to 40 tons. So it's better or outperforming Narrowcast 1, which is their lead variety at the moment with the farmers. Throughout of this, we've been engaging with the stakeholders. Your demonstration plots a little bit similar to what we heard with the cowpea, so that we're bringing as many people along with us as we can. Last month, in April, we planted 8 RNAi events, lines rather, in national performance trials in Kenya. And these ones are now going through that process where we have to do 2 consecutive cropping cycles, hoping to apply and get variety registration in 2025, and then approval for commercial release from the National Biosafety Authority as well. So we realize that we're going to have this product. It's coming, it's working. How do we get it to the farmers? So what we realize is that there is no seed system for cassava in Kenya or Rwanda, no functioning seed system. So therefore, we're going to have to build it. So we are funded to do that. We're basing it on the best and basics model that we heard about yesterday from Mercy and others, which is proven to be functioning very well in Tanzania and in Nigeria. We're adopting new technologies such as semi-autotrophic hydroponics for the EGS stage. We're now working to identify and train cassava seed entrepreneurs. This is Mama Patience in Kilifi County, who is now growing cassava, conventional cassava to start with, building this infrastructure. Where RNAi cassava comes, we will be plugging it into a system that's already functioning. And the model here is that Mama Patience and her colleagues will be growing the seed. The farmers will buy that and come back, and then you generate an economically self-sustaining system. So I'm almost finished, thank you. We're also starting to undergo branding of our cassava so we can protect it, label it, and that'll help us also with stewardship. In Rwanda, we're now growing our Event 4046 F1 lines. They're in the 2nd cycle. In Nigeria also, what we've done is we've taken this technology and we've stacked it in Nigerian cultivars, and we stacked it with iron and zinc biofortification, and those trials are ongoing at NRCRI as we speak. Products and learnings, the RNAi technology really, really works. It has exceeded expectations, definitely exceeded my expectations. We had a very defined product profile. We have exceeded that for efficacy and durability. We have 14 potential new varieties developed. MPTs are now underway. And when we're said anything like this, putting our heads out a little bit to say we are projecting, hoping, and aiming for product launch in 2025 in Kenya. All that you've seen that we did, we built the capacity all the way through. Nobody had done confined field trials of cassava before. The regulatory system was barely working in Kenya and Uganda, so we've enhanced that. These VRCA lines, RNA lines, are now available for additional breeding programs in cassava, and we're looking for opportunities to do that. And this takes time. Long-term commitment required, and I put governments in capital because bold here because we have to see the government step up. The recognition is due. These are our funders. These are all some of the people that were involved here. And I also want to shout out, we've heard a lot over the last 2 days about PhDs and training people and leaders. We mustn't forget about the technicians and the technical staff. You have to have really the high-quality technical staff in the lab and in the field to make this work. So I will leave you with that. Thank you.
So we next have the telomere story, and this story we told by Dr. Murenga Mwimali. I hope I've not butchered your name. All right, thank you.
My name is Murenga Mwimali, not the way he's pronouncing. First, before I talk, I would like to acknowledge I would like you to acknowledge my supervisor, Professor Tongona. Please clap for him. So I'll be talking to you about the current status, and basically it's about the experiences in the development of insect and drought-tolerant maize for Africa. And this is a case of Kenya. The project is called the Water Efficient Maize for Africa project, which has transitioned into what we call the Tela Maize project. As has been said here now, that it's now obvious that the world population is increasing. And the question is, who will feed the population? You can see that we are actually, as at 10 o'clock, this is the population in the world of meter. Now, I'm asking a question, how about the Kenyan case? I work in KALRO, the Kenya Agricultural and Livestock Research Organization, where we are dealing with those myriad of challenges, just like in any other space that is called agriculture. But my focus here is specific to the insect-resistant maize and the drought tolerance. As has been said before, and this is my story here, that in the English alphabet, we have 26 letters. And if you use those, depending on how you select the letters and write a letter, you can make someone dance or someone will cry, depending on which ones you pick. The issue here is the specificity that science has enabled us to understand up to the gene level. Now we know what to choose, for what purpose. Quickly, is that this is just to show you an illustration on the development of a genetically modified organism, with all the stages starting from the laboratory all the way up to commercialization. But as you learn that, it's easy to move all the way from the lab up to around here, confined field trials. But beyond environmental release, commercialization, and seed up to here, it takes a little longer than may be the case. The case of Kenya, the list that you see over there and the different stages at which the crops are indicated. and the participating institutions. My focus is about maize. How about other crops for the African population? Of course, we have different spaces and different focal points. But this means there are opportunities all over remaining. The question in the room, for you who is looking at me and looking at the screen, how many legs does that animal have? I am sure you have
backcross integration. And you are all breeders, you know, when you want good product from backcross integration, you have to go far up to backcross 4 so that you have very few background of the donor in your product. Unfortunately, we did not go for backcross 4, and unless you go from backcross 2, you do recurrent selection, taking care of quality traits, then you have a good product. But in Burkina, we just stopped at backcross 2. The product was there, and that was the mistake. Farmers start claiming for the product when the product was not really ready. But we did the trial under the control of Biosafety Agency, and we also involved all stakeholders to come and see how it was performing. And we had good data. Professor Ofori is here. You can see how statistical are talking to us. The biotech crop, that is BT cotton, was able to control efficiently 2 of the major pests. So now, what is the story after that? After that,
we want to maximize the time. When we go out for lunch, we are not going to extend the lunch break for every 1 minute. So please, let's be mindful of this. We are going to be back here by 1:30 sharp to go on to the next part. So please, if you have your questions, online, do you have any questions? Not yet. Okay. So I'm going to use my discretion. I'll begin with you from the back. 1, 2, 3, 4, Professor. Okay, so 4. We are fine. Let's begin with you. Please hand him the mic. If there are extra mics, let's hand them to the other people.
My question goes to Dr. Taylor. Nice work, but what is the plan for stewardship now that the product is going to the field?
Please, can we have extra mics?
So we're still developing that, the stewardship program. So branding our product is going to be a really important component of that. The seed system that we're building will have quality assurance built into it as well. So we're going to build it. Be monitoring the performance. Carl, our collaborators there, will be monitoring the performance in the field. So if we get any feedback, like we're seeing brown spots in the storage routes or something, they can go out and say, it's not brown streak or whatever. So we're going to have that component as well, and continuous monitoring like that. In addition, one last thing I was going to say, we are developing markers as well, so that we be able to go out and say, yeah, this is our Kingaku variety, or this is the
Francis.
Yes. Yeah, and we continue to monitor the viruses and what's going on as well.
All right. Yes, please take them. Yes. Kindly introduce yourself and then ask your question.
Good morning. Actually, it's afternoon already. Good afternoon. I'm Albert Caran from the Philippines. I have actually 2 questions. So first is for Dr. Jerry Mboyne. I'm interested to know if you'll be also employing an insect resistance management plan for the PBR cowpea, because I'm working on the BT eggplant project in the Philippines, and the regulatory system required us to actually propose an insect resistance management plan before we were released biosafety permit for commercial propagation. And I think it's also a question for all the presenters who presented about the BTA technology, because one of the main considerations in the BTA technology is the development of resistance. So that's my question, to prolong the product durability. And the question again, next question is to Dr. Nigel Taylor. So breeding for, can I? Please?
It's okay, you go ahead.
All right.
I
So
grant
it's a
you
short
that.
question. So breeding cassava could be troublesome because they take a lot of period to flower. So beyond the RNAi technology, I'm more interested to know whether you use plant breeding innovations to sort of rapidly generate your breeding populations. So such as probably speed reading techniques and double haploid technologies.
Very well. So let's have the first response, and then we have Dr. Nigel.
Yeah, so thank you very much. Yes, we have an insect resistance management package. It's part of the requirement by the Biosafety Authority before a decision is made. So we have to provide an insect resistance management package for that. But in addition to that, as I talk to you now, we have a second application before the Biosafety Authority to deregulate a second gene, the Cry2AB gene, that we are actually working on a vector stack with that to make sure that we have a second generation PBR cowpea after the first one is released, so that that one will have superior efficacy, unlike the first one, and because it has two genes, there will be issues of resistance will actually be dealt with. But beyond, in Nigeria, where it is released, I know they are doing they have a resistance package where they are mixing a bit of the conventional and non-conventional to contribute to delay resistance. The refuge in the back. So yeah, we have that kind of package there. It's part of the requirement by Biosafety Authority. Thank you.
Very well, please, Dr. Nigel Taylor.
Yep, so we did not use any modern breeding techniques. The only, maybe you could say, because we had a transgene in there and it was a single copy and we knew what it was, we did PCR, so we could discard 50% of the population straight away if they did not carry the RNAi component. Now, as we go forward, as I said, we're interested to see whether or not our product can be integrated into breeding programs going forward. Then, of course, we're going to use the breeders, and we would be using, or I would be handing to the breeders, they would be using all the techniques available.
Very well, so please, yes.
Yeah. Thank you, Mr. Chairman. My question is, I think it was partly touched on, but it was a case that the Cry1 gene in the cowpea would be, it's a single gene. And when you release it out there, it will impose selection on the pest. And the chance of breaking down is quite high in terms of a single gene. What strategies are there to make sure that that technology actually doesn't just come in and after a while susceptibility comes in again? Thank you.
So that should be to Dr. Mboyine's team. So anybody from the team could respond? Or
Okay, so I think I can respond to that question because I'm actually the program officer for the PBR cowpea coordinating the 3 countries. And in Nigeria, where the product has been released, we have the IRM plan in place. So we have the refuge. That's where you plant the PBR cowpea. We also want the conventional cowpea to be there so that the Maruca, can also feed on the conventional cowpea, and then there will be no breakdown of resistance. Secondly, for the Sample 20 that have been released in Nigeria, we don't allow them to go to the cowpea Maluka endemic area. So we're still restricting them just to the northern Nigeria where the Maruka is not endemic, while we work on stacking the gene, just like Jerry has said. And then we set up the stewardship committee that looks into, that help us to monitor, the processes. So these are what we also do to make sure that, the breakdown of resistance is not there. And then the Cry2AB, just like Jerry has rightly said, we're bringing them in place so that we'll have the 2 genes in one construct that would help the breakdown of resistance to be slow. I don't know if that answers your questions. Thank you.
Okay, so it's okay. So the gentleman.
Thank you very much. Permit me to ask two in one. One question to Gail and the other one to...
They must be very brief then. Very brief.
It's very brief. Sorry, Gail. For curiosity, or maybe for convincing regulators or other concern, is there any compositional analysis that was conducted to maybe address that? If yes, how can you compare the percentage of maybe cyanide, for example, in the transgenic and non-transgenic? And also, is it specific to a vector? No effect on other non-target organisms? That is one question for him. And the other one to Moringa. Sorry, you said there are two types of stem borer, highland and lowland. So the gene, is this specific to lowland or highland, or is it efficacious to both? Thank you.
Yeah. So that we do not have a target insect. We don't have a target, so we don't have a non-target. Yeah. And I had to go through the slides really quickly. We had 100 different components that we assessed for the compositional analysis, and there was no significant differences, including the HCN.
Okay, so that answers both questions. Please hand the mic to my senior in Katanga, brought some bias to us. We have 2 minutes, so let's finish here.
I'm in the Biosafety Committee of SARI, where the BT cowpea is being worked on. And I understood that they have a way of controlling other pests that will predispose the cowpea to the Maruca more. So by controlling those pests, the plant is in the best physical condition to still stand up to the BT, the Maruca. So I think that is one of the ways of trying to make the resistance more robust and not susceptible to breakdown of the gene. Thank you.
Thank you very much, distinguished ladies and gentlemen. When we come back, we would have genome editing in Africa. Oh, he has to respond. Very well. So, but just let's pick these pieces of information as he gets ready to respond. So the main speaker for that session will be Dr. Daniel Gigienyu, Coordinator for Genome Editing Project, West Africa, Center for Crop Improvement. So please, let's bear this in mind and come back early. So Doctor, please respond.
Yeah, the question is about the ability of the insect to, the BT gene to control the two pests. The two insect pests, that is the Busiola fusca, which is the highland pest, and Chylopatelas, which is the lowland pest, they both belong to the Lepidopteran order. And Cry1AB is specific in managing the two.
Okay. All settled now? One more piece of information will be out of here. Okay, so it's my understanding that lunch is right here. So please, let's do well to keep to time. It's exactly 12:40. We are back here at 1:30 to continue. Thank you very much. See you in a bit. Over to you.
Thank you very much. Good afternoon, everyone. Yeah, I think that would be better. Yes, I know I'm the one standing between you and your lunch, so I'll be very fast. The good news is that the keynote speaker has done about 80% of my job, so I have about 20% to do. So I'll be very quick, and since I'm doing only 20%, when you have any questions, please feel free to ask her. She'll be more than happy to answer. So I'll be looking at the overview of the new breeding technologies in Africa, specifically gene editing. And as you've heard throughout this morning, the NBTs, or the new breeding techniques, are the set of biotechnology tools that we use to precisely target and modify the genomes of organisms. And they include methods such as gene silencing, genome editing, and the likes. And they are slightly different from traditional gene modification techniques in such that they do not always involve transgenes. I said not always because the NBTs, transgenes are not their definitive characteristics. But having said that, they could still involve the use of transgenes. As Dr. Tripathi said in the morning, in the case of the SDN3s, they could involve the use of transgenes. That's why the call for regulation for NBTs. And because they are very precise as well, and they allow for the quick development of new plants and improved crop varieties. And they are not new tools. Some of them include a zinc finger, talent nucleases, the talents, RNA silencing, RNAis, and but the new kid on the block, which is revolutionizing the gene editing landscape, is the CRISPR-Cas system. That is the new kid on the block. And the NBTs can lead to precise insertions, deletions, and replacement of DNA sequencing, and then also silencing of specific genes. Now, let's go slightly into the gene editing. And again, they can induce or targeted mutations into plants effectively, and they can be segregated away using traditional crosses. And then because the crops obtained using gene editing are almost identical, or they're identical to conventionally bred crops, their regulation is waived in most cases, as we'll be looking at. And then their reduced cost also allows for efficient molecular plant breeding. And in most cases, as I mentioned, their regulatory requirements are waived, because if the plants developed using these techniques are the same as conventionally bred crops, why regulate them as if they are something different? Now, as I mentioned in my introduction, they are they were previous tools developed for gene editing, but some were very expensive, they lacked specificity, and they were time-consuming. But when the CRISPR-Cas system came on board, it paved the way for wide adoption of this use by scientists in advanced countries. Of course, just like how we lag behind the transgenic revolution in Africa, it's taking a while for us in this part of the world to adopt to these new technologies. But then scientists are beginning to get excited about it, particularly with the hope that most gene-edited crops don't need regulation. So that is, most of probably the scientists in Africa using the transgenic era were very reluctant in venturing into the technology because of the regulatory environment in Africa when it comes to genetically modified organisms. Now, this is just a quick diagram to show the difference between conventionally bred, where you make crosses, and then we go to the transgenesis, where you take a gene. The transgenesis, you can take a gene from any species and introduce into any organism you want. But having said that earlier, for gene editing, what we do is to make corrections to the existing genes of the organism. And again, as I mentioned, it could also involve the use of transgenes, but a wide application currently has to do with what? Modifying existing genes. So since there are no transgenes incorporated, that shows that the plant is the same as the conventionally bred ones. What are some of the opportunities? Now, they have the potential for increased yields and productivity, and for not just crops, but even livestock. And then this can contribute to food and economic growth of our countries. And also they offer potential for reduced environmental impact. Now, when you use these technologies, they can contribute to more sustainable and environmental friendly cultivation of agricultural practices, of crops and animals. And then, however, having said that, we need to be careful. Like every new technology, we need to be We need to take cautious steps to ensure that they are safe deployment. And it is a technology, every technology has the potential has potential hazards. However, regulations in most of the countries are already there in regulating genetically modified organisms. And it is the same, under the same umbrella that most of these countries are operating under to ensure the regulation of NBTs. What are some of the challenges leading to their deployment? Public perception and acceptance. The ground has already been muddied with the advent of genetic engineering or transgenic crops. So people are very cautious. There has been a lot of false information out there in the public. So anything that has to do with gene modification, puts people off. So there is the need for a lot of education for people to get to understand these techniques. Most of the people really oppose this technology because of lack of understanding and against science communication. The scientists are more interested in developing the products, but we were very reluctant in ensuring that people understand how these products are done, and we fail to effectively communicate to the people as to what these technologies entails, their potential hazards, and then the benefits to them. And also the regulatory environment. As we'll see soon, most of the countries are still debating on whether to adopt NBTs or not. They are trying to regulate NBTs using their traditional GMO regulations, guidelines, and this is becoming problematic for most of the countries. Now, what is the landscape in Africa? So gene editing was endorsed under the AU Agenda 2063, which is good news for us in Africa. But however, only 2 countries have implemented guidelines so far for regulating gene-edited crops. Nigeria and Kenya. Ghana, we'll see some case studies in a few minutes. But despite this, no African country has yet approved any gene-edited crop for commercialization. Now, some projects are in the pipeline. People are doing some works, but we don't have any products currently on the market. And if you can see from the maps, you see the banana in Kenya, the expert is in our midst, and also maize in Kenya, yam in Kenya, sorghum in Kenya, cassava in Uganda, cassava in South Africa, and then wheat in Egypt. Those are just a few countries in Africa currently, and I know Burkina is also doing some gene-edited trials for rice. Now, when you check the European Sustainability Agriculture through genome editing, when you check their website, we have 19 publications on gene-edited activities in Africa from 9 African countries. This database does not show the stage at which the activities are or whether they are intended for commercial release, but it just documents what activities on gene editing are happening in Africa. This is just the global landscape. Dr. Tripathi spoke extensively on this, so I'm not going to bother you about it, but this shows that the green areas are countries that are have adopted genome-edited without subjecting them to GMO regulation, like treating them like they are genetically modified organisms. The yellow ones are still under consideration, how to go about regulating gene-edited crops. And the red ones are regulating gene-edited crops as if they are genetically modified organisms. Now, the regulatory environment in Africa, we are using varied approaches. And different countries have different regulatory frameworks for approaching how to deal with new breeding techniques. And it ranges from strict regulations to more flexible approaches. However, there's the need for more clarity and guidelines from regulatory bodies. We need to be more consistent on how we go about the balance between biosafety and innovation to promote public and acceptance and participation. Because if the regulatory bodies seems to be, treating these organisms very, very, differently, obviously going to put public fear into people. People are going to see these technologies as a monster. And for that matter, the public will fail to readily accept products from these technologies. Now, let's just look at the cases of some of the countries that are currently having either regulations in place or are drafting regulations for NBTs. Now, for Nigeria, they have approved guidelines for gene editing. And they are the first African country to do so using a science-based approach. Now, when you go to GMOs, one of the reasons why GMOs failed in most countries is that people are rather regulating the process instead of regulating the product. When you regulate the product, it's much more easier because what is a GMO? When the eventual product has a transgene, you can classify it as a GMO per some of the definitions. But when you intend to look at the product using how it was developed, obviously you eventually class that product as what? A GMO. So they are using science-based approach. Do we have a transgene in the plant or the product? No. If that's the case, then it's not a GMO. So they are using more science-based approaches. Yes, Kenya has also published their guidelines on gene editing. And again, the research and products will be viewed on case-by-case basis. South Africa. Interestingly, South Africa has the longest history when it comes to GMO regulations and commercialization in Africa. But interestingly, South Africa is treating new breeding techniques, or they are regulating products from new breeding techniques as GMOs, which is very worrying, particularly since you are more like the pace setters when it comes to gene editing. But they are currently handling issues of NBTs as a GMO. So any gene-edited crop currently in South Africa is classed as what? A genetically modified organism. For the case of Ghana, Ghana is currently developing guidelines for gene-edited products. And the case for Ghana is not different because Ghana is also using a science-based approach. So what the current draft is proposing is that products from gene-edited crops, if they do not have a transgene, they will be classed as what? If they do not have a transgene, they will be classed as what? A non-transgenic crop, and then they will take a different regulatory approach. So that is good news for Ghana. For the case of Uganda, Eswatini, Burkina Faso, they also are currently far advanced when it comes to developing guidelines for NBTs. Now, when you come to Mali, Senegal, they are also currently either having regulatory systems in place or they are planning to release one. And then the challenge with some of them is that they are contemplating whether to adopt the GMO system for regulating gene-edited crops or to use a different regulatory approach. And we are hoping that they will also use a more of a science-based approach where when you don't find a transgene in the product of a gene-edited procedure, it's classed as a non-transgenic crop. What are some of the factors that are influencing? Some economic factors. We need to look at the cost effectiveness and accessibility of some of these NBT, and because this can have a direct influence on adoption. and also the political will. If the political environment is not ripe, no matter what we do, the technology is not going to get anywhere. And also the societal influence. Once people understand the science of the technology, and they get to understand that the product is developed for the benefit of man, but rather to the detriment of man, I believe it will go a long way for the... So there's the call for a lot of public education and public awareness creation. Now, in the director's introductory remarks, he mentioned that WACCI is planning to become a hub for genome editing for the sub-region, and we are in conversation with our partners from the Danforth Center and also UC Davis and other partners to ensure this dream becomes a reality, so that we can have scientists from the sub-region coming to WACCI to train as experts in gene editing. As a way of conclusion, NBTs have the potential to revolutionize agriculture in Africa by addressing food security, improving yields, and also mitigating climate change. And careful consideration of the regulatory, ethical, and safety concerns is essential for the responsible deployment of what? NBTs. Collaborations, investment, and research are critical in harnessing the full potential of NBTs, and also realizing their benefits for agriculture and livelihoods. And to finally add, just as we missed the transgenic revolution, we should tread cautiously so that we don't miss the gene editing revolution as well, because we sat on the fence waiting for what others would do, and then before we started jumping on board. As my colleague from SARI mentioned, Ghana released its first transgenic crop gave approval for environmental release of our first transgenic crop just this year. This is sad. Despite the technology has been around for the past 2 decades, it took us several years to get to where we are. We are all encouraged in Africa to ensure that we don't wait on the fence to see this new technology that has the potential of alleviating food and nutrition security on our continent to go by. So we are all encouraged to ensure that we all become advocates for this technology, to ensure that, because this is the continent where the technology is needed most. This is where it's needed most, and we don't have to wait for posterity to judge us for allowing our kids, our sisters, our brothers to die. Would they rather die or get solutions so that they don't blame us for doing nothing? So we are all encouraged to take action to ensure that we tap into this new technology so that we can have a food secure continent. Thank you very much.
It's my understanding that we can do one more presentation before lunch is ready. So let's have Dr. Valentine Tui, who's a research scientist at IITA, Kenya, to come speak to us on genome editing strategies for developing disease-resistant banana. In the meantime, if you have any questions for Dr. Jijenyu, please get them ready. Okay, so we'll take the second presentation and then we'll take those questions. Hopefully, we'll still have time before lunch.
Yeah, good afternoon. So I'm Valentine Tui. I work with Yelena Tripathi. We are based in IITA Nairobi Station. So this afternoon, I will be talking to you. Actually, it's more of science-based than just the general talk. The strategies that we are using to develop banana for resistance to diseases, but specifically for resistance to BXW, banana xanthomonas wheel. So introduction, I will not go to this because Lina already spoke about this. But let me emphasize a little on this. So you see the importance of banana. You can even look at the leaves. Probably if we go out there, if there are no plates, we can just go into the bush, cut banana leaves, wash them, and use as plates. So you see how important banana leaves are. And you can also see, hey, where is the pointer? I hope many of you know this, and probably you have eaten that. That is cocky. Banana leaves are used for that. You can even use it to tie some things. Those the packaging, especially during Christmas, during Valentine's, and so on. So that shows you the importance of banana. So, but the most unfortunate thing is that despite all this importance, the banana is having a lot of constraints, specifically diseases. This slide was shown in the morning by Lena, so I will not emphasize. But what I am going to say is we have actually developed resistance to this disease. We have actually knocked out this, and we are also trying to develop resistance to this. So my presentation will be focused on this disease, bacterial wilt. So conventional breeding could be used, but the problem is that it has a lot of challenges. Most of the cultivated cultivars are triploids, and so they are highly sterile, making pollination very, very difficult. And so one of the things we can use is biotechnology. And when I mean biotechnology here, I refer to genetic engineering, which is the classical genetic engineering, and genome editing. And so, but the purpose of this talk, I'm going to focus mostly on genome editing. So, a brief definition of what genome editing is all about: targeted and precise changes in the genome of an organism resulting to gene modification or mutations using molecular scissors called nucleases. And so, these nucleases, there are different types of nucleases that have been developed. And so the nucleases do what? Induce double-stranded break. So we know that DNA is double-stranded. And so when these nucleases, what they do is they cut the double-stranded break, and then the cell repairs itself. During that repair mechanism, some of the nucleotides are lost, a few are added, or even there is base replacement. So we have different types of tools that are used for this purpose. We have zinc finger nuclease, we have talent, we have CRISPR, and one of the tools that has made gene editing to be so pronounced is CRISPR. But before CRISPR came, there was zinc finger nuclease, there was talent, there was even meganucleases. But because of the robustness of CRISPR, how it is very efficient, very easy to use, many people started knowing what gene editing is all about. And of course, Lena mentioned in the morning that the people that got Nobel Prize chemistry were actually those that developed CRISPR tool for gene editing. And so in our laboratory, we are actually using CRISPR and not the other tools. So again, apart from CRISPR, there's also another tool that is being used. You have base editing. Actually, all of these, either base editing, prime editing, they're also based on CRISPR. But the issue is that CRISPR cuts the DNA, creating double-stranded break. But this does not. So what happens is that with this, a deaminase is fused to the CRISPR. And so when deaminase is fused, the CRISPR loses its nuclease activity. Instead of cutting the DNA, what happens is that it flips just one nucleotide, depending on the deaminase that is fused to the CRISPR. So in the case, we have two types. We have adenine deaminase and cytosine. So you can see from here, cytosine deaminase can flip a G to an AT. And so that is what happens. And then we also have prime editing. In this case, it uses a reverse transcriptase enzyme that is attached to prime editing RNA, gRNA. And then what happens is that when it is attached, it forms this loop and then cuts the DNA on one side, and then when the repair mechanism happens, you have some base pairs that are mutated. So in our laboratory in Kenya, what we do is we are using two methods. We are using the knockout, and we are also using CRISPR activation. I will explain CRISPR activation later. So for knockout, we had to actually establish the system first in the lab, because we did not just go, we didn't just want to go straight into knocking out some genes. We had to establish the system. And so for us to establish the system, we decided to first of all focus on PDS, phytoin desaturase. So this is an enzyme in the carotene biosynthetic pathway. So what this enzyme does is that when it is mutated, the plant becomes albino, white. So that chlorophyll is lost. So and this is all, in many labs, this is used as a marker actually to establish gene editing system. And so we decided first of all to focus on this. And so in banana, there are two genomes, A and B. And so that is always the challenge. So most of the genes are duplicated. And in most cases, the genes are not the same. So you can see, like this is the PDS gene. In genome A, see how big it is. And in genome B, see how small it is. And so our duty is to make sure that we try as much as possible to mutate both the gene in both genomes in order to have 100% mutation. So we try to target the conserved regions. So we design our guide targeting the conserved regions in genome A. You see where the guides are in exon 5 and 6, and in genome B in exon 2 and 3. And then we made this construct, and of course, delivered into the banana cells. And as I told you, when that gene is mutated, the plant becomes white. Look at the nature. You can see the white color here. So this is already a practical example that the system is working. So when she showed in the morning how we created disease resistance, probably some of you might be thinking, but then in this case, you could really see that CRISPR is actually working in banana, because we have been able to do what? To mutate the PDS, and the plants actually lost this green color, and most of them are white. And then we check whether this thing we are seeing is not as a result of the selection marker. And so we decided to do what? To sequence. When we sequenced, we saw that there was mutation. And so the next thing was, we also decided to knock out DMR6, which Leonard showed in the morning, but I'm not going to discuss this because she already showed that one in the morning. The next one was L-nodulin. So this L-nodulin, you know that nodulins are found in leguminous plants, but there are some cases where they are found in non-leguminous plants. And so when Lena did transcriptomic analysis, RNA-seq, so she discovered that this gene was highly expressed, was highly upregulated in the susceptible cultivar as opposed to the resistant. And so we saw that if that is the case, we could try to actually mutate it and see whether if we knock it out, we are going to have some resistance activities. And so we focus on that. We knock it out. When we knock it out, we saw that the plants were, this is the wild type, and these are our mutants. The mutants were highly resistant. And so that means this gene is actually playing another role. Instead of nodulation, it's also playing another role, which is what? Disease resistance. And so another method we are using is CRISPR activation. So in CRISPR activation, what happens is that the Cas9 is fused with an activator. And so when that is done, the Cas9 also loses its nuclease activity. It doesn't cut the DNA. Rather, it helps to promote, to activate the expression of a gene in question. So what we did was we fused the Cas9 with an activator. This is the activator. And so when that is done, the Cas9 does not cut the DNA anymore, but then it will activate the expression of endogenous genes. So we produce, based on the transcriptomic analysis that Lena also did, we realized a couple of genes that were upregulated in the resistant cultivars, and some of them were downregulated in the susceptible cultivars. And so we decided to focus on the genes that were downregulated in the susceptible cultivars so that we could do what? We could try to upregulate them, activate them, so that they will be able to confer the resistance to BXW. So what we did, we now produce 6 different CRISPR activation constructs targeting different genes. We did this and then delivered into the banana genome. And when that was done, we regenerated the plants. And so the issue was, were the genes actually upregulated? Were they activated when we did use this CRISPR activation method? You could see from the qPCR that most of the genes were upregulated. So that means this system is actually a good system to actually activate the expression of some genes in the plant. And then we also check whether this activation we are seeing could result to resistance. We subjected the plants to inoculation with Xanthomonas. Again, as you can see, the wild type is having the disease. And those that were mutated, we did not see the disease. Yes. So we see that the wild type is having the disease and the others are resistant. So that shows that the CRISPR activation is actually a good mechanism for developing resistance to diseases. And we look at also, this is the disease rating. Look at the wild type, it's very high. So that means we have actually used 2 systems. One is the knockout, and another one is activation to actually confer resistance to BXW. So we have published a couple of papers. If you really want to know the science and the details about these 2 technologies we have used for banana, please, you can contact some of these papers. They are there. This one is in yam. So we did not just work on banana alone. We have also done that system in yam, where we actually knocked out PDS in yam. So these are our papers. They are there. All of them are open access, so you can get them at any time without paying any money. So this, I will have to acknowledge, this is our team in IITA, the sponsors, root tubers and bananas. And of course, probably we are going to have this at lunch. I hope you people have done that. Thank you.
Right on time, right on time, right on time. Please turn on the light. Thank you very much, Dr. Valentin. Okay, so this is what we are going to do. Our guests will be ushered downstairs. I think Barbara and her team are going to ensure that. The rest of us would pick our lunch from the corridor. You can choose to eat on the corridor or come back in here and have your lunch. We'll resume this session at 2:15, and we'll have a few more presentations before we close. So, Barbara, please take the guests to where they will have their lunch, and the rest of us, please, let's go to the corridor, pick our lunch. Feel free to use the corridor or come back in here and eat. We'll see you at 2:15. Your choice. No, Dina, you don't have to eat at dinner. We can go have a conversation at Kempinski. Yes, anybody wants to take a good guess? What exactly I would have told this young man? Remember, this was 1999. I didn't have one, no. No. Yes. 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 Last one. Say again? Okay. All right, so my response to the young man was, I left it at home. Now, truth is this. I had heard of email addresses at the time, but I didn't have one. I didn't know what they were. Now, 2 months after this incident, my friend who was in Lome at the time created my first ever email address, busyforsomething@yahoo.com, brought it to me with a password, and taught me how to use an email. Fast forward into the early 2000s. I entered KNUST at the turn of the millennium when the whole Y2K conversation started. Those were the days when the people who came from Accra to Kumasi, KNUST, were the ones who had computers. And if you say computers, those big ones with a hunchback. So if they were coming from Accra, they actually bought seats at the back of a bus and put them there and brought them to campus. And it felt good to have one computer, not necessarily in your room, but on the floor. I mean, so a floor of about 25 rooms. If one room had one computer, you could actually walk to your lecture theater and go brag about it, go tell your friends, on the same floor where your room was. Then at some point, so those are the days we were saving our works on floppy diskettes. Then at some point, pen drives came around. Very few people had pen drives. You see the Odipo is research scientist at the National Crops Resources Research Institute in Uganda. He will be taking us through this. Please, let's give him a round of applause.
I would prefer to have this. Okay. Hello. Hello, good evening. Good afternoon. You know, the good thing of presenting after lunch is that the timekeeper and the MC can sleep, and then they will not be timing you. And also the good thing is that my mentor, Nigel, already presented about cassava. And my first supervisor, also at MSc level, Lina, already presented. So I feel very happy to use the same podium with them. Yes. So my presentation is going to be a simple one on improving cassava through gene editing. But I decided to insert CRISPR-Cas-mediated gene editing because that's the commonest approach being used. Yes. So I will take you through introduction to cassava, but dwell more on what has been done with gene editing in cassava before ending with summary. Actually, for your information, the person standing on the right is me. I'm very close with cassava, and that's how I've been throughout my life, and I feel happy that I'm talking about it here. So I will not talk much about cassava introduction, but what I can tell you is that cassava means livelihood in many African countries and the tropics at large. It's the source of food and income for many farmers. So you will not remove cassava from a number of families and institutions in Africa. It has many applications, and it's now moving away from just being a household
commodity to becoming a very important industry commodity. And because of this, predictions have been made and identified cassava as one of the most important commodities that will survive in the climate change era. And therefore, for that reason, it's important to increase investments in cassava. Growing cassava is not just without problems. Some of the problems, Nigel already talked about it in terms of diseases, but I will go back and let you know that also in addition to diseases, we have climate change related calamities that may set in the future. So in the meantime, a number of strategies are used to continuously improve cassava so that the end users have options. So among them includes crossbreeding, which someone already talked about. There's mutation breeding and transgenic breeding. But I will start from the option of genome editing, which has already been exhaustively introduced. Cassava has mainly relied on, to date, the option of gene editing that has been used is CRISPR-Cas based. And so it's important to know what it takes to do it. The most important thing one needs at the start is a good quality reference genome sequence. It's this sequence that helps you to design guide RNAs. The guide RNA is the 20 base pair sequence that guides the Caesar, the called Cas. It can be Cas9 or whichever type of Cas. It guides the Cas to the region of the genome that you want to make changes in. So once the constructs have been made, they're delivered using different approaches, and after you have generated your plants, you can check them for the type of mutations that have been introduced and later evaluate the phenotypes. So it all started in 2014 when I got an offer to do my PhD research under supervision of Nigel Taylor. Before that, you will just read about genome editing in other commodities. So when we started, the simplest thing to do was to use a visual marker gene, phytoindesaturase, which was talked about earlier on. So I used the same gene and made guide RNAs, which were directed to the 13th exon. And after constructs were made, these constructs were transformed into cassava using the approach reported earlier. And I generated cassava, which, for the lines which were expressing the guide RNAs, the plants lost the green color, which was a confirmation that the gene editing complex that we introduced successfully made changes in the genome of cassava. And this was already a sign of success. So apart from just looking at the plants physically, I was able to confirm through sequencing that indeed there were changes in the targeted region of the PDS gene. And so, which is very important for the cassava brown streak causing viruses to cause infection. And what they found was that plants that had 2 of those L4 genes edited showed high level of resistance to cassava brown streak disease. And so feel free to get additional information from the publication. So another group also published work on using gene editing to target cassava mosaic disease causing geminiviruses. So they targeted 6 of the genes within the virus, and they found that in terms of phenotype, there wasn't much difference between the edited plants, lines, and the ones that were not edited. But all the same, an attempt was made to develop resistance to cassava mosaic disease using gene editing. Recently, another group in the US used zinc finger nuclease mediated approach to target a promoter region of sweet-tail gene. This gene is very important for the bacterial blight pathogen. to cause infection. And so they were able to see improved resistance. So these were some of the few cases where gene editing has been used to attempt development of resistance to cassava-related diseases. And also, effort has been underway to develop cassava which can resist commonly used herbicides. Why herbicides? Because cassava production involves several times of weeding, and unfortunately, it's mostly women who weed. A good number of men leave it to the women, which is so burdensome. So the idea here is, can we have cassava which expresses the resistance to herbicides, and therefore when we spray the weeds, the plant remains unaffected but still yields highly. So that was really done, and the control plants die, but edited plants continue to do very well, and if it was under field environment, we will still obtain very good yields from the edited lines. So these are some of the progresses, and in addition, efforts were made to also look at quality attributes. I said that cassava is grown mainly for storage roots, but in the storage roots, there's a lot of starch. So that starch has both amylose and amylopectin. So in the starch industry, some types of starch are easier to process than others. So, but in this particular case, cassava with high level of amylose was developed so that industrial players will take advantage and produce as much as we need for our starch industry. So efforts are going on to incorporate this. Yes, thank you. I'm glad you are, you are tracking me. Yes. So this work has been published, and we can also here look at how cyanide deactivation has been successfully done in cassava by two groups actually. One group was from Kenya and another one in the US. They have both demonstrated that if you target cyanide biosynthesis gene, you can produce edited plants with reduced level of cyanide, and therefore this has implications for producing cassava which is healthier for humans to consume. So also as part of my PhD work, using gene editing, I was able to identify genes associated with flowering, but particularly I was able to use gene editing to deactivate anti-flowering genes, and in so doing, lines of cassava that had edits in the targeted anti-flower genes were able to start flowering from the petri dish in the tissue culture, yet under field environment they will take up to 8 months to flower. And whenever I planted those lines in the soil or took stem cuttings from them, they retained the ability to flower. The implication here is that you can use gene editing to switch on flowering in cultivars that have never flowered, and therefore open opportunities to increase the parental base. And also in addition, the flowers produced here can be used for breeding, because I made crosses using flowers and I was able to produce very nice fruits, as you can see over there. So this manuscript will be going out soon. So another group has already ventured in developing double haploids. It is hoped that when we have inbreds in cassava, it will improve cassava breeding. And when I came back from US, one important thing for me was to see if what I did the other side can be translated into success in Uganda. So one of the constructs for PDS was successfully used with the Timson there, and we were able to produce cassava that has edited PDS, and beyond this, it hasn't been easier to do a lot more work because the funding became an issue, and a few times when I applied for grants, I wasn't successful, but I still keep trying. All right. Challenges. Cassava is not straightforward to work with. Nigel said at some point, and he knows, what happens is that almost each cultivar is a different story. So what it means with gene editing is that you have to keep reinventing. If you want to work on a different line, you have to optimize. And so this is a serious issue. And also with gene editing, the efficiency is one thing that needs to be worked on, because there are still off-target issues where sometimes a few genes, you can get cases of off-target effects as in general for gene editing. So another, oh no, the point, okay, finally, what I want has come. So another point is that in Africa, generally, I think we still have low human capacity that's capable of doing gene editing practically. And in addition to having fewer human beings who can do that, there's limited local funding capacity. Most of the work that has been presented in this house, I'm sure over 98% has been funded by international donors. You see how that leaves us open to doing nothing beyond just talking. So I really find it challenging. And another thing is, while we do this science, regulations is still an issue. We saw how some countries have regulatory regimes and others don't have. So that's one thing that is also a challenge. Yes, I'm getting there. So in summary, you can see that cassava has already become applicable to gene editing applications. And we have seen only one successful report of cassava gene editing out of the many I talked about. And many trials in gene editing of cassava so that we have very efficient system. I strongly end by saying that after this kind of very good debate, for us to have meaning, our own African institutions and the continent at large needs to customize gene editing by allocating significant budget for biotechnology research in general, but gene editing in particular. And if we were to have any meaningful gain from gene editing, we shouldn't do it in isolation. We still need to work with other teams so that we're able to generate products made in Africa by Africans, and therefore we can have gene editing revolution in Africa. So I wish to thank the organizers of this meeting. My supervisor, Nigel, is here. Part of this work is what I did under his supervision, and also my Ugandan NARO supervisor, Alisai Titus. This is where I end my presentation. Thank you for listening.
Thank you very much. So our next presentation is on improving sweet potato through genome editing. And this will be taken by Samuel Achampong, who is a research scientist in the Department of Molecular Biology and Biotechnology in the University of Cape Coast, Ghana. You have 10 minutes, sir. Thank you.
Good afternoon. I feel honored to be given this platform to share what I've been doing with fellow scientists across the globe. Today I wish to speak about improving sweet potatoes through genome editing. My name is Samuel Achampong. I teach and I also research at the University of Cape Coast, the University of Choice in Ghana. Just a brief introduction. The Millennium Development Goals. One of them is that government should try as much as possible to eradicate extreme poverty and hunger. That was to be achieved in 2015. We are in 2023, and I don't think this goal has been near realization. There are still about 45% of deaths attributed to poor nutrition. Still, there are about 45% deaths among children under 5 being attributed to poor nutrition. Add it to the rice and give to the children, because that's the only thing she has. So the rice will be sweet, they will eat, but you can guess that these children are going, the belly will be full, but they are not getting the required nutrient for proper growth and function of the body. And that is what we call hidden hunger. You are not getting enough micronutrients for your body. According to some reports, globally there are about 1.5% deaths that can be attributed to vitamin A deficiency. 1.5% of deaths attributed to vitamin A deficiency alone. Now, if you add iron deficiency, anemia, and then the deaths resulting from iron deficiency, that's a lot. And even if the percentage is just 0.1 or 0.001, this is preventable death. So we don't have to allow these things to happen, because we can do something about it. Ghana and many other nations have made great strides to improve the situation. However, there was still more to be done. Currently, it is reported that about 21% of children are still deficient in vitamin A. There are different interventions that are used to supplement nutrients. So we have school children being given folic acid, because at this stage they lose a lot of blood and they need to form hemoglobin to replace what is being lost. We also have the vitamin A supplementation. This was very successful. We also have fortification of the food that we eat. So in Ghana, it is mandatory that the cooking oil that we use should be fortified with vitamin A. So we have Fritor. We have, then we have the salt that we eat is iodized. Iodine has been added. The wheat product that we consume, they should be, fortified with some nutrients. But so a study carried out indicates that the locally made wheat flour has very poor amount of iron. Now the figure there at the right is the red color indicates which was imported from Turkey compared to the locally made, which is blue. You see that about 4%, 4-fold increase in iron content in what is imported from Turkey. That means Ghana, we are not doing something right. So there are different ways of fortifying food. You can add it directly, or you can also breed to improve crops, so that the crops will produce the micronutrients, so that when you eat, you get those nutrients. So what we want to do is to replace the supplementation on the left, giving the capsules. We are no longer giving the capsule, but we are going to give food that has enough of the nutrients that the children require. That has been the idea. Yeah, Uganda has some, an excellent intervention. The government provided cows to families. So if you have cows, you get milk, and from time to time, you can also get meat. That's another intervention. Biofortification of food crops has advantages, because mechanism to improve this, that would be very good. Unfortunately, there's a linkage between high dry matter content and beta carotene content. So that means it is nearly impossible to separate these traits. If you have white variety, you are going to have high dry matter content. If you have orange flesh variety, you are going to have low dry matter content. So it's difficult using the traditional method to achieve this. So we want to supplement these efforts with new technologies such as genome editing. A lot of the colleagues, my senior colleagues, have talked about genome editing. I won't tell much about it, but one thing I want to emphasize is that genome editing is more widely accepted as compared to the previous biotechnology approaches, because there are two mechanisms you can use, what we call the ribonucleoproteins. The proteins will enter the cell to edit, but very exciting. So in order to do genome editing, you need to study the pathways, look at the trait that you want to achieve, and then study, there's pathways. So for improving beta-carotene, there's a long process. I won't talk about that, but there is one of the genes, beta-carotene hydroxylase, that converts beta-carotene into other downstream products. And because of that, in many varieties, the beta-carotene doesn't stay. As soon as it's produced, it's converted to other products, so beta-carotene doesn't stay. So if you can have a way to block or reduce the activity of the beta-carotene hydroxylase genes, then a lot of the beta-carotene is going to stay, and you can have sweet potatoes with orange flesh color. So I started the work at North Carolina State University. There were challenges with the work, particularly with regeneration. So in genome editing, you are editing a cell, and you need to get a new plant coming from a cell, an edited cell. So there's something called somatic embryogenesis. I won't talk about that now. Somatic embryogenesis, generate a callus, and then you get a plant from a callus. And in sweet potato, that is extremely difficult. So we have optimized the process. And then there's a different method, organogenesis, and that seems to be working for us. Sorry. So the picture on the right, that's for organogenesis. So you can see new shoots coming out from a leaf. This is through organogenesis. So this is what we have achieved so far. I was working on two genes. Beta-carotene hydroxylase gene, and then cell wall invertase inhibitors. The cell wall invertase inhibitors was more important in the US, so that work has been more advanced. But in Ghana here, we are interested in the beta-carotene. That's what I'm working on. And there are some regulatory issues, other issues, but I won't talk about that now because of time. I have been signaled. I want to say that we have the technology, we have the men, we have what it takes. What we need is some support, financial support and other support to do what we must do. Thank you very much.
Thank you very much. So the last presentation before we take questions will be on CRISPR gene editing of maize, experience and prospects for Africa. This will be taken by Dr. Elizabeth Njuguna, former postdoctoral research fellow, International Center for Genetic Engineering and Biotechnology in South Africa. Please, yes, come over. Please put your hands together for Dr. Njuguna.
Good afternoon. The introduction has been done, so I don't need to repeat myself. I'll go directly into the topic that I've been invited to, but I'm also very grateful to Alliance for Science and WACCI for this invitation. It's really an honor to stand in front of many names that I just read online. I've read a lot of your papers. I'm really happy to be here. Thank you for the invitation, really, and I look forward to knowing more people along the way. So I'll start, like many of my predecessors, with a brief introduction on why we need to do research on maize. I know Dr. Murenga already talked a lot about it. So it's a staple food for at least around 300 million people in sub-Saharan Africa, probably even more by now, and a source of income and livelihood for many smallholder farmers. Maize is grown in at least 43 million acres of land in 46 out of 54 African countries, at least from the search that I did recently in the FAO statistics. And this area is still increasing as the population grows. Currently, we are producing our yields at very low, at least 2 tons per hectare, which, if you compare with the global production rate of 5.8 tons per hectare, we have a lot of work to do. We have a lot of improvements to do. Also, our maize is largely rain-fed. The production is largely rain-fed, and this means that it's susceptible to the effects of climate change, the droughts, the floods, the salinity, and so we also need to work on that. And some of also, indeed, pests and diseases, climate-related and other pre-existing ones, like the ones mentioned here already. The picture on the right is a smallholder farmer field in Nakuru, which is my hometown, so it's a pleasure to show you. Nakuru, Kenya, sorry. So a little bit more about facts on CRISPR gene editing for maize improvement is the CRISPR technology has rapidly expanded gene editing in maize. If you see, there's now more papers, there's more research groups that are involved in maize gene editing and the transformation work as well. So this is good for our future. And also there are currently reports of DNA-free gene-edited maize. You know, unlike the humans and the animals, crop gene editing has taken longer before we can get transgene-free plants. And so we have already like one with the herbicide tolerance that is completely transgene-free. Maize is also one of the few crops that has been edited using all the 3 site-directed nuclease approaches that Dr. Lina quite well explained, so I wouldn't have to go to it. And also there's a recent breakthrough in the transformation of maize, which is really great, that previously we had this recalcitrant, difficult to transform maize plants, and also elite breeds that could not be directly transformed, and sometimes we would have to start off with breeds or with varieties that have a lot of undesirable traits, but this is no longer the case. By using morphogenic regulators within the transformation construct, we're able to induce somatic embryogenesis and also shorten the tissue culture step, because the callus induction step is also skipped, which is really a great invention. So now I'll jump directly into part of the work that I did during my PhD, and this involves the modulation of energy homeostasis in maize to study drought stress tolerance. I must say that different approaches were used. This was on the very last leg of the project that I did, the CRISPR part, thanks to previous regulatory challenges. This work was done in a collaboration between the VIB Center for Plant System Biology in Ghent University and the Plant Transformation Laboratory at Kenyatta University under the guidance of Professor Stephen Runo. The picture there, I'm in between my transgenic maize plants at the lab in Ghent. I had to be selfish. The metabolic engineering need, we have 3 key genes of interest: the poly-ADP ribose polymerase, the poly-ADP ribose glycohydrolase that reverses the activity of the poly-ADP, and also the NADX hydrolase. All of this, no, sorry, I lie. The PARP and the NADX are some of the ones that we worked on, but on this work, I'm just going to dwell on the PARP gene. So on a normal day when there's stress, or other DNA damage within cells, which occurs regularly, PARP is one of the first respondents, and what it does is that it uses, it takes ADP ribose molecules from this energy costly NAD, NAD nicotinamide adenine dinucleotide, we all know it, and it adds ADP ribose chains into proteins, several proteins, transcription factors, histone proteins, and also on some of its own, and once it arrives, it triggers DNA repair process. During stress, there's an increased production of the reactive oxygen species. That means the whole, the pathway is more triggered because PARP activity is highly increased, and the energy costly NAD, to replenish it, we need a lot more ATPs. So that means the energy levels in the cells are quite decreased, and plants in general can only stay or cope with energy levels they only have a very small window within which the plant will eventually die. So the idea here is, can plants be more efficient in stress response? Can they be energy conservative or use energy more better? Previous studies have shown that RNAi silencing of PARP gene in Arabidopsis and Brassica enhanced stress tolerance. In Arabidopsis, we see this is a wild-type C24 plant, and when the PARP was knocked out with hairpin silencing, and also the PARP2 knocked down, we already see some good response to the drought stress that was induced. Also, the Arabidopsis, Brassica, we can see a transgenic plant that has a PARP2 gene knocked down, and it's already responding better than the azygous or the control plant. Here now, I'll jump into the CRISPR part of the project that we carried out. We used the agrobacterium system of transformation. This is a tDNA construct that we developed, and we had two guide RNAs. One was driven by the rice ubiquitin promoter, the second one was driven by the wheat ubiquitin promoter, and then the Cas9 nuclease driven by the maize ubiquitin promoter. You know, you hear ubiquitin because within the monocots, we still have a limited number of toolbox, the promoters, so many ubiquitin works well, we go with it. And we have the cauliflower 35S promoter that we used for expression of the Bar marker gene. And we had three constructs. The first one was targeting a big deletion within the maize PARP2 gene. The second construct was targeting also a big deletion, at least 200 base pairs within the maize PARP1. And the third one, we targeted either frameshift mutations with one guide RNA in each of the two PARPs. I must say we looked for homologs of the Arabidopsis PARP genes in maize, so there was two of them that were of interest. Sure. On our outcome, we saw that the T0 shoots, we had 8 out of 20 that had the big deletion of at least more than 200 base pairs. And then for the PARP1 gene, what we have were more of either frameshift, indels, the 80 inserts, homozygous, and all 4 base pair deletions within the maize PARP1. For the third for the third construct, interestingly, we had wild-type phenotype, meaning that none of our guide RNAs worked. Even for the second one, this guide RNA didn't work. All our mutations were within the first guide RNA. So we went ahead with the PARP2, and what we did was we selved the lines with big deletions, at least 4 of them. And here is, again, an analysis of the Cas9 nuclease deletions, and we see that all the plants had maintained their big deletion, but also we were seeing new inserts or small deletions within the transgene, meaning that our initial plant was not a homozygous deletion within the big deletions. There could have been wild type. Sorry, can you hear me? Yeah, there could have been wild type phenotypes that were masked earlier. We also did a Burgeon segregation analysis of these ones, and we were quite interested in these plants that were showing, that were showing negative for the Bar marker gene, meaning that it's possible that the tDNA was already exited, and this would be interesting plants that have Cas9 deletion, but with no transgene. I'll quickly jump into some of the preliminary functional analysis that we carried out. I must say preliminary because we all did it in the T1 lines that I've just shown on this table. And the first one was a DNA damage assay that we conducted. For interest of time, I'll quickly jump through and just say it was a paper roll assay. Whatever is as indicated here, we allowed the plants to grow and introduced hydroxyurea on the 5th day of growing, and then measured the root length daily. 12 seeds were grown per genotype, per treatment, and we see the outcome. We had 4 CRISPR lines. The background in which the work was done was on a B104 wild-type plant, and we also had a mutant plant that is already known to be hypersensitive to DNA damage. The blue graphs show the controls that were not induced by the DNA damage, while the hydroxyurea-treated ones are orange. So what we noted is that the mutant line behaved as expected. Wild type was also reduced. The root growth was highly reduced. Yes, but we had 3 CRISPR lines that were showing some tendency. I can't say it's final, and this was honestly not significant, but we were expecting, because this is a mutation that is not yet stable. So we hope that if we do similar analysis on T2 and T3 CRISPR lines, we may see some interesting phenotypes, and also maybe including more larger samples in the assay. The second functional analysis that we carried out was a water deficit assay on this T1 PARP2 mutant lines with a big deletion. It was done on an automated watering, weighing, and imaging platform at the VIB Center for Plant System Biology. 2 conditions were used: the well watered and the water deficit. And the robot tried to maintain these conditions on an everyday by watering up to the water moisture content that is expected. And we had 7 genotypes per plant, per genotype, per treatment. And these plants were allowed to grow for at least 4 weeks, after which we measured several endpoint parameters, which are summarized on these graphs. I won't quickly go into them, but I'll describe that the darker green ones show the well-watered, while the light green shows the water deficit in all the graphs. And in all the graphs, again, the first bar is the wild type, while the 3 are the CRISPR lines that we had the deletions. What we notice is that there are several endpoint parameters that show a trend of tolerance and a water deficit, but this still also needs to be done where we have a stable and uniform mutation. And probably also someone indicated that our stress was a bit less severe, and we need to introduce more severe stress because it induces different kind of pathways upon stress. Finally, I think this has been mentioned, so I don't have to go keenly into it, but the other gene editing projects happening in Africa on maize is the development of maize lethal necrosis resistant hybrids. A quick mention is that it's interesting when it's being done on hybrids already and trying to maintain on an already existing trait. Sorry, that confused me. So the one other thing that is quite interesting is that the seeds will be produced royalty-free, and the companies that enter into commercialization agreement with for the commercialization of this will not be allowed to sell them at higher cost, which is really, really great. Finally, my last slide is that I'll mention some of the prospects that CRISPR gene editing would be useful for Africa. Some of these are work that is being done out of Africa, but here we have, it can be used for enhancement of the nutritional content. We've seen a lot of slides before coming in on what these either essential amino acids, minerals, or vitamins can be done. It can be used for development of climate-resilient maize. We have already seen that these water-efficient and energy-use-efficient approaches, but maybe other approaches can be used. The more the better and the merrier. And also for more pests and diseases, because maize lethal is just one of them. There's many more. As well, we can use it to increase the yields of maize and hopefully also have shorter growing cycles. Why should we wait for maize for 6 months or 8? And development of herbicide-tolerant maize, that's already happening elsewhere, but we can do it for us and energy-use-efficient ones. Yeah, so I think I'll leave the last part because these are benefits that have been mentioned over and over. Thank you so much for your attention, and yeah.
Right, time for questions. 1. Okay, please pass the mic. 2 and 3. In that order.
Thank you very much for the wonderful presentations. My question goes to the first presenter on cassava. He talked about using genome editing to knock off cyanide in cassava. And I know cyanide is associated with resistance to certain pests, particularly the whitefly. So won't this activity subject or make the plant prone to attacks by some of these pests. How do they handle issues of susceptibility that arise due to knocking off some of these defensive mechanisms that a plant may naturally possess? So as you move towards desirability by humans, you make them more susceptible to attacks by these pests. So I want to know how they are trying to manage the two.
That should be to Dr. John Odipu, right? Okay. Please, let's have him respond quickly. He stepped out? Oh, he's here. Yeah, give him the mic. Let him respond.
Yeah. Thank you for your question. Yes, I talked about knocking out genes for cyanide biosynthesis. What I didn't say is that it is not complete elimination of the gene expression. What happens in the process is that a few base pairs are altered or deleted with some institutions, but it basically keeps the gene partially functioning. It means the cyanide role, which has been important in cassava, will still be played, but there will be some reduction in overall expression, and that is what will reduce the content of cyanide in the edited plant, but still the plant will be able to function. Yes.
Okay, very well. Second question, yes.
So my question was to the sweet potato in Ghana. So you're proposing to knock out the genes downstream of beta-carotene production, but the products of that are zeaxanthin and bioalexin, which are important photoprotective agents. If you take out that pathway, you are likely to be negatively affecting photosynthesis.
That is right. So sweet potato has 6 copies of the gene, so we want to reduce the amount. So we are not blocking it completely. There will be some production, but we want to reduce. We want to reduce the rate of conversion of beta carotene.
But that presumably could still affect photoprotection if you're reducing the amount of the downstream components.
Yes, please. Yes, please. Yes, please.
Thank you. So, third question.
Thank you very much. Well, my question is one to cassava and even maybe the sweet potato. Anybody can help maybe in answering that. For the cassava, they tried gene editing to deactivate anti-flowering genes. So, for example, if I have a crop, maybe a legumes, cowpea in particular, and we have a problem of photosensitivity, what do you think, based on the maybe experience or the literature maybe you read, can we also deactivate a photosensitivity gene so that at least the issue of this response to day length maybe overcome? That's the question. And then, for the last presenter, I just need clarifications. You made mention one of the T1ZM PAR1 or PAR2, one of them, say CRISPR lines shows tolerance to DNA damage. Was that intentionally maybe designed to do that? If yes, then tolerance to that DNA damage, what is it? What is it done? I mean, what do we get from that? Thank you.
All right, so please pass the mic. Let's get the presenter to respond to that before we take the last question. Where's Dr. Elizabeth? She stepped out? Oh, she's there. Yes, let her respond.
Should I start? All right.
Yes.
Great. So DNA damage, why we were interested in it is that the poly-ADP ribose polymerase is known to be a fast respondent normally during DNA damage, and it's also important for oxidative stress. So why we did that assay was to actually see, once we have introduced the DNA, once we introduce the DNA damage, how are the plants responding? I showed phenotypes where we were testing the root growth. Within the roots, we have a higher cell division, and that means there's also likely to be more DNA damage during the high frequency of cell division. So the plants that were highly susceptible, like the mutant plant we used, you see already the root length reduction was already much lower because it's already hypersensitive to DNA damage, while the CRISPR edited plants, we expected that they will be more tolerant. That means the plants, the roots will not be, the root length will not be as highly reduced because the plants are tolerant and they're able to survive better. I hope I answered your question. Thank you.
We'll take. Okay, yes.
I request you to ask that question again. Something about the.
You attempted gene editing to deactivate anti-flowering gene. Yeah, so what I'm saying is, how applicable is that maybe method in other crops? For example, cowpea. We have a cowpea that maybe is high biomass and also grain yield and large seeded, but it's photosensitive. What do you see if we deactivate the photosensitivity gene? Will that work maybe to get?
Yes, thank you. A number of crops are sensitive to day length. Like in our case, cassava is from South America, where the change in day length is nearly almost negligible. So what happens is, although it responds to some degree to changes in day length, when you play around with some of the genes involved in flowering and evaluate the products under the neutral condition, which is the same thing I did, you get results which represent any condition where cassava will be grown in the world. But in your case for cowpea, it's true that some commodities or some crops are sensitive. In order to know whether it is day length causing that kind of behavior in growth, or whether it is maybe expression of some genes, maybe if it is possible, you may have to collaborate with some colleagues and expose the cowpea to varying length of days. That is maybe longer days, longer sunlight period than night periods, and you see how it behaves. But also, although it may be a bit costly, but through collaboration it is possible. You can also just study gene expression to see which genes are expressed more in this variety under your conditions, or maybe you can study gene expression under conditions of long day or short day or day neutral, and then you will be able to tell. But right now, I can't tell. But what I know is if you manipulated some of the genes involved in flowering, you may also get some result. But right now, the answer is not straightforward. Yes.
All right, thank you very much. We have two more questions, and then we'll move to the next presentation. So, yes, and then the gentleman here.
Thank you. My name is Zara from Burkina Faso. My question goes to Elizabeth. For the knockout of the gene in maize for drought, she said that there are a knockout of a gene that intervene in the uses of, I think, is it the carbohydrate synthesis or the drought tolerance? Because I know that maize is a C4 crop already, and it has a natural resistance to drought. So if you knock out, it will not affect the carbohydrate synthesis in Calvin cycle. Yeah, that is my question for her.
Thank you for the question. I don't know about the carbohydrate synthesis. I didn't mention about the carbohydrate synthesis. The PARP pathway that we were targeting is because the PARP gene in itself is known to be a respondent to DNA damage, normally even during normal cellular processes. But then its activity is hyperactivated during stress, and it consumes the large of the cell's energy. So that means the plant is actually dying because it's over-consuming its energy unnecessarily, a bit unnecessarily. That's what we expect. So what if we just reduce the expression? And the best case scenario for us is we don't completely eliminate all PARP activities. We knock down one and retain one because we still need its function in DNA damage response or activation for the same. But then we want to reduce the overuse of energy.
Okay.
Yeah.
Thank you.
Very well. So last question to my right, and then we move on to the next set of presentations, please. Please pass the mic.
Thanks. My question maybe goes to area presenter, Rina or Tyra, especially on the cassava and banana. These are vegetatively propagated crops. How are you working with seed company, and how are they going to be effective? Because maybe over time farmers can keep giving each other the foundation seeds, and end up maybe having other problem with resistance. So I needed to know how the seed system would protect. Thanks.
Okay, so Lina want to respond to that. Please bring the mic.
Yeah, so as banana is a vegetatively propagated crop, so we basically work with the tissue culture. Means, okay, we are not there yet, but we are already involving tissue culture companies. We are providing some trainings to them, so that they will be the one who will, for deployment of the seeds at that. But we will not stop farmers, if they want to get the what you call the seedlings from their friends or from the neighbors. We will not stop them. They can still go ahead and take that from them. But first, the mass multiplication and deployment will be through the seed company. And as Nigel mentioned in his presentation, we'll have the same thing, so that means the material will be developed through the public institution, so that means there is no royalty or anything attached to that. But then if farmer goes to the tissue culture company, they have to pay to buy the first seedling, but at the cost whatever is the tissue culture seedling is, not adding any additional cost because of the royalty. I hope I answer your question, right?
Very well, thank you very much. So we are about 15 minutes behind time, so we're going to try to manage the rest of the day. My timekeeper, please be very exact with when you should prompt the presenters. So we have 3 more presentations before we do a panel session. What can Africa learn from the UK's biotech experience? Biotech crops in Latin America and lessons for Africa. Agri-biotech in the Philippines, replicating the best practices. So in this order, Professor Gilles, okay, let me take a deep breath. Professor Gilles, please put your hands together for him. Please, you have 10 minutes.
I didn't know I had the hardest name to pronounce. It's Giles Aldroyd. Eric asked me to give a perspective on what lessons we can learn from genetic modification or biotechnology in the UK. And just before I start on that, I actually want to just say what a fantastic meeting it's been and what a very timely meeting it's been. What I think I've taken away from this meeting is how many products are so close to actually getting into the market and having impact. And I feel like we're at the beginning of a wave of biotechnology that's going to sweep across African agriculture with very positive impact. But I think it's really important that the community in this room pay attention to not only solving problems for farmers, but paying attention to the appropriate delivery of those technologies such that we don't get a backlash against that wave of biotech that's going across Africa. Because that would set us back 20 years if we do have a massive backlash against that biotechnology. We have to handle it very intentionally, knowingly, with our eyes open. So what can we learn from the UK? Well, I tried to find the best way to describe the state of GMOs in the UK. And I would say that pretty well describes GMOs in Europe. It's not been a good situation, as I'm sure most of you are very keenly aware. The position in Europe and still within the UK is very anti-GM, and actually, probably certainly in certain parts of Europe, that is strengthening, it's not weakening. I think in England, it is sitting on the fence at the moment with regards to biotechnology, and things are changing. But if you were in Austria or even Germany, it's a strengthening position against biotechnology, not a weakening position. And you know, when that is a result of the general public taking a perspective. Now, I'm probably not the best person to dissect why we're in this situation in Europe. I think the original handling of the technology by Monsanto in the early days was very poor, and they were very insensitive to the position of Europeans for corporations and the sense of having technologies foisted upon Europe. That was not very good. But I think for us scientists, we also allowed the debate to be controlled and set by certain NGOs with vested interests. And we really lost that debate. Now, as a result of that, of course, with technology, it was a new technology. We're talking about 20 years ago now. It was a new technology back then, and we were all learning. But unfortunately, we in Europe are suffering the consequences of that learning, and still suffering the consequences today. So I do think that Africa has some very serious lessons to learn about what happened in Europe, and to ensure that you do not recapitulate what we experienced 20 years ago and is still having ramifications for us today. And that's recognizing that when you're releasing these technologies into the market, although you're targeting farmers, and farmers are going to be very welcoming of those technologies as they were in Europe, it is actually the public that you need to be paying attention to. Is the public behind you in the release of those technologies? And that's something very important to consider. Now, 20 years ago, we didn't have the Alliance for Science in the UK, we didn't have the Science Media Centre, and these organizations have really come about in part because of this reaction within Europe. So I think we're in a much stronger position now, because we have organizations that are helping us as scientists reach out to the community and reach out to the public and to contribute. Importantly, we as scientists contributing to that debate and not being presumptuous that everybody's going to accept these technologies. So I think for Africa, you're in a much better position because of what our own experiences in Europe, but do this with your eyes very wide open, and don't allow the debate to be controlled by certain NGOs. Now, I mean, the situation in Europe is pretty poor. There is one GM crop, the BT maize, that is cultivated in Europe. So there are a few countries. The dark brown are countries that ban that GMO. So it's intentionally banned from production in all of the dark brown countries. They cannot grow that particular BT maize. And in the light brown, so Portugal, Spain, Czech, Slovakia, Romania, have some low-level cultivation of BT maize. The biggest cultivation by far is in Spain. So it's not that Europe is GM-free. Most people think that Europe is GM-free. It's not actually GM-free. There is BT maize produced in Europe, but it's a very restricted production. And that is the full extent of GMO production in Europe. So contrary to where we are at in other parts of the world. Now, as a British scientist, you know, we have a very, very strong base in plant sciences in the UK. And it has been extraordinarily frustrating to be sitting in this situation, because in the UK, I'm pointing out, these are all products that we have been developing in the UK. So we have purple tomatoes. In fact, Kathy Martin, the John Innes Centre, has a whole range of tomatoes with higher vitamin D, higher anthocyanin, higher carotene. That's a combination. This is a GM product, but she also has gene-edited products. The new vitamin D tomatoes are gene-edited. These have been around for a very long time. They are not commercialized in the UK. They're being commercialized in America. And in fact, I believe you can now buy GM purple tomatoes in the US. It's extremely frustrating for us as scientists to be sitting in the UK where we're actually developing products for other people and other continents. Jonathan Jones, very effective blight-resistant potatoes. They're carrying 3 transgenes from wild potatoes that give very effective very effective and stable blight resistance. Again, these are being commercialized in America. They're not even close to or far from ever being commercialized in the UK or anywhere in Europe. We have field trials going on for high iron and zinc content in wheat. That's happening at the moment. Those are looking really effective. We have camelina lines that are producing fish oils. That's all working beautifully. And we in Cambridge are now running field trials with barley, getting hypercolonization, colonized by beneficial fungal associates. And all of this work by all of these British scientists, some is ready for commercialization, some is showing very promising results in field trials. None of it's going to be available to a British consumer in the near future at all. In fact, all of that commercialization is very likely to be happening in America. Now, that's an extraordinarily frustrating situation for us to be as British scientists. I've got to say, thankfully, the British government has not stopped research in this area, despite the fact that its commercialization is challenging within the British landscape. So we as scientists can still get funding to do this kind of work, but it's very frustrating just to see it all commercialized far from our shores. Now, you've seen this image multiple times, and I actually think I've got the one that's a couple of years old now. But the point here that I really want to make, not to reiterate what Lena already highlighted on this slide, but that in fact the position on the anti-GMO position in Europe has really extended to an anti-gene editing position, and the majority of Europe takes a very hard view on gene editing, that they must be regulated as GMOs, and therefore this is not only stopping genetically modified products from being developed, it's also stopping us from using what is truly a revolutionary technology of gene editing. Now, my perspective as a European scientist, I consider myself European despite Brexit, my position as a European scientist here is that it's really holding us back. It's holding us back decades. We're 20 years behind the curve now in Europe. And have this wonderful tool of gene editing and to not really be able to see its application, really a very frustrating position. So I want to just sort of highlight here a cautious tale. It's we as scientists can get really so excited about all the potential of these technologies, and we've got all of these products. Please make sure that we do it in a very intentional way, work with organizations like the Alliance for Science, and do it very thoughtfully. Make sure you bring in governments and the people along with you, because you do not want to be in the position where we are in Europe, where we are developing all of these technologies, and we will, as a society, will not be benefiting from them. Now, I dislike Brexit intensely. It is not part of my politics, but as a plant scientist, we're probably the greatest beneficiary of Brexit. And that's because, so what happened with Brexit when we left the European Union, we imported all European law. So your law, in much of British law over the last 30 years, has been created within Europe. And so you can't just stop. That law is intrinsic to your system. So what we've done, we left the EU, but we brought all EU law into British law and made it British law. And then, which means including the GMO position. Now, interesting, the regulation in Europe, regulation exists for GMOs. It's not that GMOs are banned, they are regulated. And in fact, the regulation, when you look at it, is actually reasonable. It is the interpretation of that regulation that has been the most challenging situation in Europe. A proportionate application of that regulation would create an environment where you are able to release GMOs in Europe. But the interpretation of law is people, and of course, people or politicians are responding to their populace. So you can have regulations in your country, and I would say the regulations we have in hand would theoretically allow the commercialization of GM products in Europe. But the interpretation of those regulations is extremely conservative, that makes it impossible to actually go through the process of deregulating a GM product for commercialization. It is the interpretation of the regulation. But because of the consequence of Brexit, we have imported the GMO regulations into the UK. But very importantly, and this just happened for us about 6 weeks ago, this is the passing of the Precision Breeding Bill, or the genetic technology. We know it as a Precision Breeding Bill, which I think was very insightful. Precision Breeding Bill basically says that gene-edited crops that lack a transgene do not have to be regulated as if they're GMOs. Also, interestingly says that basically says anything that could be achieved by conventional breeding should not be regulated by a GMO. So it also means cisgenics. If you take a gene from a wild potato and put it into a conventional potato, providing there's absolutely no foreign DNA associated with that, then that would also fall within this regulation of not being considered a GMO. So at the moment, principally, it's really gene editing, but it could also be utilized for cisgenics. Now, this is a fantastic news for us. So we were already able to do field trials with gene-edited crops without having to go through the amazing regulation for GM. Now we can actually commercialize gene-edited crops within the UK. And it really sets us apart very significantly from the rest of Europe. Now, I think something important to say at this point is that what happens in the UK is really influencing. So Europe is paying attention. My understanding is that they take a very extreme view at the moment with gene editing, but they may be forced to step back from that extreme view. And certainly the movement in the UK, it's also happening in Norway, who are part of the EU, but have a slightly different relationship that means they can regulate differently. Those movements towards promoting for gene editing is also influencing the environment within the European Union. Now, I think one of the really unfortunate things about the European position on GMOs and gene editing is it's really frustrating for us as plant scientists sitting in Europe not being able to commercialize those products. But I think it's extraordinarily unfortunate how much that debate and the position that Europe has taken seems to have influenced the position in Africa. And I see that changing dramatically now, but I think the last 20 years, African governments have been looking much more to the European Union than they have perhaps to America, and taking as such taking a very cautious approach. So this ever so slow melting that started in the UK, that might, I hope, continue through the EU, I hope will also remove some of the barriers of the oppositions that you might experience or might be experiencing here in Africa. So the Precision Breeding Bill is really opening up opportunities for us within the UK to start really pushing on gene editing, and certainly puts the UK in a very competitive position for developing gene-edited crops in a European context. If we presume that the whole EU will eventually have to accept gene-edited crops, then the UK is in a very strong position for developing those gene-edited crops. So in Cambridge, we've been developing the Crop Science Centre. This is an initiative I've been leading. It's really about taking all of the discovery science that we are driving in plant sciences in the Cambridge landscape and pushing it into impact, stopping it sitting just in Arabidopsis and demonstrating what is feasible. Now, what I hope that we can achieve at the Crop Science Centre is some truly transformational type work, and we're focusing on replacing or eradicating the need for organic fertilizers, using beneficial microbial associations, enhancing the efficiency of photosynthesis, reducing losses from pests and pathogens, and some very innovative technologies that we hope will increase the efficacy of plant breeding. Now, I just want to touch on this because talking a little bit about my own research and where I think we could be in the next decade. So this is not technologies that are going to be available in the near future, right now, but I'm hopeful that we can be developing these technologies in the next wave. And what I'm really interested in are technologies that cut across this interface between the highly mechanized industrial farming systems and that of a smallholder farming system. I think most of the technologies that we've heard about today were developed in this context of farming, but clearly have applications over here. And it's simply the fact that the drive or the capability to move those technologies from these sort of high-income countries to low-income countries has been really limited and slow. So I'm really excited about more of those kind of technologies that really push agriculture globally rather than just benefiting high-income countries. So the vision that we're working towards is a situation where we can deliver most of the limiting nutrients that we currently apply through inorganic fertilizers, to deliver those through our crops, through beneficial microbial associations, so enhancing interactions with arbuscular mycorrhizae that massively help in the uptake of phosphate and water, transferring the association of nitrogen-fixing bacteria from legumes to cereals. And this is what we are doing within the ENSA project, these underground associations that are so beneficial to plants. And we work very, very closely with our sister program, RIPE, which is increasing photosynthetic efficiency, driving up increasing the efficiency of carbon capture. And if we can stack these traits, we really have the potential to be truly transformative, to be really break the current system of high input system, but also drive up productivity massively for smallholder farmers. And just to explain that a little bit further, this is both of this, the arbuscular mycorrhizal fungi, they're carbon auxotrophs. Every single molecule of carbon they get comes from the plant. So it's actually, in many ways, you should think of this as, it's very much an extension of the plant. It just happens to be fungal hyphae. But those fungal hyphae have been built from photosynthesis in the plant. The energetic reaction of breaking this triple bond that sits between the molecular dinitrogen, it's incredibly energy demanding, and you have to break that bond, you're using energy from photosynthesis. So you have to energize these systems, and if you don't focus on energizing those systems, then you take yield penalties. But by combining increased photosynthetic efficiency with this, then we actually can overcome those energy requirements and drive productivity. We reckon we need about 20% of photosynthates for these microbial nutrient services. RIPE's modeling says they can increase photosynthetic efficiency by 110%. 110%. So I take the first 20% of that, and then there's 90% left that can go into, or 80% left that can go into more production. So if we can achieve this vision, it really truly is potentially transformative, not only for sustainability, but for actually raising productivity overall. I think I'll take 30 seconds. Just the basis of engineering this association is not completely out of the blue. We're not starting from scratch. Nitrogen fixation, when it evolved, it didn't evolve out in isolation. It actually used, so this is where nitrogen fixation evolved about 100 million years ago, restricted to a group of plants in the angiosperms. All of the genetics, or most of the genetics that was used at this point didn't evolve at this point. It evolved all the way down here at the base of the land plants associated with the arbuscular mycorrhizal symbiosis. So that many of the genetic frameworks that nitrogen-fixing bacteria utilize are also utilized by the mycorrhizal symbiosis. And a key example of that is this symbiosis signaling pathway that allows plants to recognize nitrogen-fixing bacteria, but also allows plants to recognize arbuscular mycorrhizal fungi. Now, so because of this framework, we've been able to engineer the perception of nitrogen-fixing bacteria. And this is, I'm showing you here the signal transduction pathway in action. And this is not in a legume. This is in our engineering model, barley. And these are responses to the bacterial signaling molecule, nod factor. So cereals can recognize these bacterial signaling molecules because they're utilizing a signaling pathway that's already there, present in cereals. Furthermore, we're able to induce or engineer nodule-like structures on barley. This again is in a cereal, it's not on a legume. So we're really getting there. We really can, but it takes much more to get than just getting a nodule-like structure on a cereal. We actually have to get the nitrogen-fixing bacteria into those cells of that nodule fixing nitrogen. So although over the last 10 years of work we've come a long way, we've still got a ways to go, and it remains something of a moonshot to get to nitrogen fixation, but one that I'm optimistic about. In my last slide, I just want to highlight, that this is a sort of transformative technology. It's not available today, but we hope it's available in the next decade, that we'll be getting to that place. And both the RIPE and ENSA project have been adopted by this new initiative, Bill and Melinda Gates Agricultural Innovations, which is a spin-out from the foundation focused explicitly on product development. And it's really pushing both of these projects, my own and the RIPE project, into really thinking about products, how we get those products, and how we actually really start to have the impact that this vision entails. So with that, if I can just come full circle back to my question, which is what can Africa learn from the British experience? Well, firstly, I'm happy to say we're still able to do fantastic science in plants, and we can deliver really effective products for the world. And that, I think, is a good thing. But I think it's a cautious note. Don't get too focused on just the impact and the products and farmers' products. You have to pay attention to society and politics, because that is really going to dictate how successful your products are going to be.
I guess I have to fire my timekeeper. All right, so we have two more presentations quickly. Let's have this light. Thank you. Let's have Pablo Orozco from Global Policy, from Alliance for Science, who is a Global Policy Director. You'll be talking to us about biotech crops in Latin America and lessons for Africa. Yes, Pablo.
Right, thank you. Pablo Orozco. It's great to be me, the one whose name gets butchered. It's a nice change. So, hi everyone. The good thing about being one of the last speakers is I can bank on all the wisdom that we've gotten throughout the day. The bad thing is I'm going to have to make extra sure you're not asleep in the back. So I'm going to do that I'm going to talk about the experience the Alliance for Science has had on policy development for genome editing in Latin America. But my true objective is to walk you through the process of establishing new regulations for genome editing. And here's the kicker. I want you to think and tell me the answers you would give to regulators asking you questions that I've gotten during this process, and I want you to think what you would respond. So that's what I'm going to do. But before I do that, I want to thank WACCI, Professor Dankwa and Joseph Opoku-Gakpo for the invitation and for the wonderful job that they've been doing, not just today, but in the past years. So we're short on time, so I'll skip the Alliance for Science mission, but I will focus on what I think is a common thread throughout the day. Ultimately, if I got it right, if not, you can tell me right now, everyone here wants to leverage the knowledge of our local scientists to achieve local solutions to the problems we face, right? I dare you to tell me that you don't want to. So, and we've seen this throughout. We know Dr. Lina Tripathi does an excellent job of this, but we know that it's not simple. It's complex, and it covers these three pillars that we talk about when we discuss the innovation and enabling environment. And the thing is that they're interconnected. So what I'm asking you to please think about is what role you play in those. Which of these are you helping build up for Ghana? Which of these are you helping to build up for, to build up for Africa? Because I want to see Africa, Latin America, and Asia work together to build these up and to learn from each other. So, I'll go quickly through this one. I think we've talked about so when we think about genome editing, we have to think about what are the issues that come up. And ultimately, the discussions have been many, but as many of the previous speakers have said, it ultimately comes down to the question of what oversight do these products need, and do we apply the GMO regulation to these products? Now, nothing happens in a vacuum. When our countries begin to think about how they regulate this, they don't start from scratch. They take a lot of things into account, which I'll go into, but one of those things is, what is the current national biosafety systems? What do they say for GMOs? And all of their, not just concepts and principles, but think about the general position that countries have towards technology. Quickly going through a summarized version of the process, we start with what we have. What concept principles do we have? So we have to take into account national biosafety system, government policies, and general country positions. Then we have a national and international discussion on how we should approach these new technologies. And what we need to take into account there is usually relevant international agreements, concepts. How does a country's biosafety system define a GMO? That is key, that we're going to look at in a second. And then, what are scientists doing? What's the level of public participation that scientists have in defining policies? And ultimately, we end with the actual creation and implementation of either directives, regulations, or laws. And there, it can get messy. We need to decide what agency has oversight, what type of regulations. Do we use a law? Do we use a directive? What do we use? And then we have to think about the regulatory trigger. So I'm going to try and apply my own teachings on science communication and not get too technical. But please talk to me after if there's something there that you want to expand on. And ultimately, the judicial branch, because as Europe well knows, the results of their decisions at court will define their approach to genome editing. So we've all seen the slide. We should give it a prize for the one most used. So I won't go into it. But what I'm going to try and do now is give you a overview of what Latin America has done so far in genome editing. But here comes the first question that I'm going to ask everyone to answer, right? So the first thing I think I'd like you to think about is, what do we even call this technology? And it was great that your past presentation, because you added one to the mix. So we're going to evaluate 4 options. We're going to say genome editing, new breeding techniques, plant breeding innovations, and just good old CRISPR-Cas. So who wants to call them genome editing? Show me hands. Nobody wants to call, okay. New breeding techniques. All right, we got some takers. Who wants to call them plant breeding innovations? And who wants to just use CRISPR-Cas9? Nobody. Sorry, CRISPR. Okay, so that's a point. So all of the Latin American countries have focused on NBTs, right? And please think about this. If you're in a meeting, if you're at an international event, and some regulator comes to you and asks you, okay, new breeding techniques, genome editing, when we talk about this technology, is it one technique or is it many? I know you can all answer, but think about how you would answer that in 5 minutes flat, because that's all you're going to get. These different countries, which are the countries in Latin America that have directives or regulations on genome editing, took the approach mostly of using new breeding techniques. I think only Brazil used precision breeding, but most of them, for example, Guatemala and Honduras also use genome editing. So that's the first thing. What do we call it? Second, which is a really important, is what institutions has oversight. So if you look at the chart, I don't know if you can see it very well. I apologize for that, but I'll save you the time. All of these countries have focused the oversight on the Ministry of Ag or the Ministry of Science and Technology. Now, I'm sure that you've worked in this space enough to know what that means. Next, what is the regulatory trigger to say if an end product is a GMO or gene editing? Now here I want to cover two things. One, I think, direct thing that we can learn from Latin America is that they knew their audience. They knew we pay attention to environmental agreements, and they knew that the Convention of Biological Diversity and its Cartagena Protocol has power. And so they focused everything they proposed in trying to define a science-based approach. They highlighted the fact that their definition of GMO is the same one used for LMOs in the Cartagena Protocol. So doing that, when Argentina first set their regulatory approach to these this technology, they banked on that. And they were also take into account that Argentina is considered a center of reference for biosafety since 1996. So that also gave them a push to be bold and try to set science-based regulations. And so all of the countries followed the same GMO definition. And in that definition, the key or the regulatory trigger is, I'll read the definition of GMO because I have another question for you, just to make sure you're with me here. A GMO is a living organism that possesses a novel combination of genetic material obtained through the use of modern biotechnology. I'll stop there. That definition, would that give us an approach for a regulatory system based on product or based on process? Who says product? Who says product? So that definition could be used for product-based regulatory system. Who says process? And what do the people that didn't lift their hands say? Right. Well, that was a trick question because that talks about both, right? It talks about the novel combination of genetic material. That's based on the end product. If the end product has a novel combination of genetic material, okay, then we're looking at the end product. But that definition also says obtained through the use of modern biotechnology, and that is a process. I know. Okay, so this is, you all know ultimately Argentina's approach to genome editing is a decision tree where you basically say this is GMO or this is not GMO. And the criteria is if there's a novel combination of genetic material, there is not a novel combination of genetic material, if there's no foreign DNA, if the edit could be found in nature, and if it could be produced through conventional or traditional breeding methods. Now, the wording is different, but that's the ultimate outcome, which is followed in most of the countries in Latin America. So quickly going through this, all of these countries did not issue new regulations. They used their national biosafety systems definitions and just added specific guidelines for genome editing. And they made the clear distinction between gene-edited products for agriculture and for animal. So that's first takeaway. Second, their definitions were compatible with the Cartagena Protocol. That was big. Watching these different countries establish their regulations, you could tell it made a big difference. We've talked about that they all establish a procedure to define if a product is or not, does or not fall under GMO regulations. And this is interesting. Some of these countries establish a procedure for developers to ask the regulatory agency if their products in their pipeline could be considered as a GMO or as a, could fall under GMO regulations or not. And I think that's really important because that's looking at what we discussed, the enabling environment. They really want to push developers to have open communications channels with the regulators to get to the end, which is getting the products out. So for example, there, Argentina, Brazil, Chile, and, pipeline, yes. Argentina, Brazil, Chile, and Paraguay have it. Colombia, Honduras, and Guatemala doesn't, although talking to the Colombian regulators, they say that one of the things they've learned from the US and other regulatory systems is they need to have an open communication with developers. So they're trying to do that unofficially, but that's just between you and me. Unofficially, it says it doesn't. The other thing I want to mention, I know I'm out of time, but list. I would advise against putting lists on anything that is regulatory because then you box yourself in and you're left with this list for the next 10, 20, 25 years, and this technology keeps evolving. Only Brazil has a list on the techniques that they accept. This has allowed Latin America to look for harmonization efforts. One example is Guatemala, El Salvador, and Honduras, who have a system where they don't only allow gene editing, but they're allowed data transportability that hasn't begun to function yet, but the first stepping stone is there. The last thing is Brazil and the rest of the countries for Mercosur are trying to work on other ways that harmonization of regulation can happen. And I promise this is the last slide. Oh, wait, no, that was a lie. This is the last slide. I'll end with this. The conclusions are, we need fit-for-purpose definitions for our regulations so that we can have a biosafety system that is functional. And the word functional there is key because functional means your products will get through the regulatory process and into the hands of the people who need them the most. That is the one key takeaway I want you to take. And I want to say that the Alliance for Science is not just working on general in Africa or in Latin America or in Asia or in the US or in Europe. We really, really want to bank in collaboration. This is the last slide that we did a side event at the last Convention on Biological Diversity, and the key takeaway there was that we have safe and locally developed GMOs that both African and Latin American countries are developing and that we can be proud of. And I think that sentiment of us having our own tools to push our countries and communities ahead without damaging the environment is such a powerful thing that it really moved heads there. So thank you so much, and I'm sorry for the extra time.
Thank you very much. So we now have Albert John Karan from the Institute of Plant Breeding, University of Philippines, Los Banos.
Hello, good afternoon. Are you ready to hear from an Asian boy? All right. So probably a lot of you have been wondering what am I doing here in Africa being an Asian boy. Spanning from 13,000 miles or kilometers from Accra, Ghana to the east. And so today I want to take a different path of explaining how the Philippine biotech systems work in the Philippines for us to be able to generate products for the farmers and the consumers in a more timely manner and progressive manner. And so for this afternoon, I'll be presenting the Philippine biotech systems and how African countries can replicate the best practices and overcome the challenges that we in the Philippines have faced for so many years already. So again, I'm John Albert M. Karan. My background is in horticulture and molecular biology. And I'm currently a research and development officer of the Biotech project in the Philippines. And also, I am a global fellow of the Alliance for Science. So let's get into it. So first, why and where is the Philippines? So I've mentioned already, so the Philippines is somewhere here on the east side of the world. And we are directly adjacent to Ghana, to East and West Africa. And so our climates are pretty much similar. So it's also humid. And I think that's one of the reasons why me, as an Asian counterpart, sharing to this audience what the practices we have been doing in terms of the Philippine biotech sector. So, similar to Ghana, we are a tropical country, but the main difference is that we are an archipelago composed of more than 7,000 islands. And so we also face different challenges, and one of those challenges is ravaging of the 20 plus typhoons every year. And probably that's the reason also why the Philippines is very welcoming in terms of the innovations that is relevant for improving the lives of the farmers by making more resilient farming communities. So now, I'm going to provide you with a brief overview of the Philippine biotech and biosafety. So for context, this is the Philippine government policy statement on biotechnology. So let's read. We shall promote the safe and responsible use of modern biotechnology and its products as one of the several means to achieve and sustain food security. Equitable access to health services, sustainable and safe environment, and industry development. So as you can see, it's pretty positive in a way that it was crafted way back 20 years ago when the then president acknowledged the importance of biotechnology in improving the agricultural sector in the Philippines. So in fact, the government has also instituted an annual National Biotechnology Week where we expose or where we show different biotechnologies developed in the Philippines. So it's being held every November. So now let's take a look at the biosafety side of things. So I won't go into details of all the things that are happening here, but this is I just wanted to show you this because this provides an insight of how dynamic and co-evolving the biosafety framework of the Philippines, because we don't have a law that says that yes, you should regulate these GM crops, but we do have biosafety committees that ensures that we are regulating products of modern biotechnology in a progressive manner. So we don't want to be restrictive, and that's why the biosafety regulatory system of the Philippines have been evolving and evolving and evolving. So I just want to point out one thing that is very relevant to the current discussions. So in 2021, we released an actual memorandum circular to not regulate products of gene editing. So those that are coming off from SDN 1 and 2 are not going to be regulated as GMOs. But if it comes to SDN 3, insertions of more than 20 base pairs will be regulated. So yeah, that's it. And in 2022, we actually adopted a new system for regulating GM products. So for instance, the Biotech project, we were the first public sector product that applied to the new regulatory system, and it only took us about 7 months from the application to the approval of a commercial propagation permit. And so, for some key achievements, because of this regulatory system that is being applied in the Philippines, so what are the key achievements that we have so far? So 129 total GM events have been approved in the Philippines. So these are mixed with the imported for food and feed and processing, and with the approvals for direct use as food and feed in the Philippines, and also for field trials and commercial propagation. And we currently have 3 GM crops approved for commercial propagation in the Philippines. These are namely the GM corn with stock traits, the golden rice, which has been a recent rave in the world because this is the first biofortified rice or carbohydrate source in the East Asia. And finally, the more recent one that we just had commercial propagation permit last October 2022, the BT eggplant. And recently, we also, the regulatory body in the Philippines also recognized the one application from Tropic Bio, the banana that is non-browning. So they recognize that this is not going to be regulated as GMO. So I believe they are now going on to the field trials or the performance trials for the non-browning Cavendish banana in the Philippines. And for me, I think the regulatory system has encouraged this kind of culture of approval process in the Philippines because it will not come through, the products that were regulated will not come through to the market if not properly regulated by the system that is being in place in the country. So in the Philippines, so as I mentioned, we only have the regulatory system, the gene-edited products will not be regulated as GMOs. So to further explain how these approvals were made possible, I will now share to you what the biotech systems in the Philippines look like. So the biotech systems in the Philippines, so for us to be able to understand who are the key stakeholders, so they are the academia and tech developers, so state universities, colleges, public research institutions, and private industry. And the government and regulatory sector, the biosafety committees, plant and animal bureaus, and of course the funding agencies. And lastly, the end users and the civil society sector, farmers, consumers, and civil society organizations. So, okay. So now, let's dive deeper to the academia and tech development sector. What are the best practices that are being made so that the regulatory system will work the way it should be working? So first, the best practices in the academia. So we have a responsive higher education curriculum. They are revamping the educational curriculum every 2 years, so it should be up to date and relevant to the current advances in the modern biotechnology space. And there's also an increasing number of research centers that conduct modern biotech products. So in the past years, the Philippine government invested a lot in the Philippine Genome Center for the aggressive collection of data sets from a lot of natural resources. And there's also a huge investment in the crop biotechnology centers, fisheries biotechnology centers, and animal biotechnology centers. And that's separate from the state universities and colleges research centers. So we have been expanding a lot in terms of the research institutions that conducts modern biotechnology research. And there's also a very much collaborative research environment between the public sector and the private sector. So we have a functioning collaboration with private sector, and just like in the BT eggplant project, we have an existing partnership with an Indian company. So that's considered a South-South partnership and a public-private partnership. So also there's an emerging startup culture for the development of biotech products. So I think there's about 50 startups in the Philippines that deals with different biotech products in the agricultural sector, healthcare, etc. So the downside on that is that there's still a long time of research. We spend a lot of time from research to product release or the market release. So because of excessive bureaucracy. And another challenge that we see in terms of the academia and tech sector is the increasing workforce competition, because there's a lot of influx of new graduates. There's a limited tenured positions, especially in the public sector. And lastly, there's a gap between the biotech research and the social sciences. We see this because there's a lot of increasing oppositions to the NGOs that are more into social action. So we see this as an opportunity as well, not just a challenge to overcome, and then in the future probably to create more interdisciplinary research projects that involves the social sciences as well. So now let's move on to the biotech systems in terms of the government sector and the regulatory sector. So what are the best practices? So first, institutions have strong capacities on the regulatory sciences and risk assessments. So because I said earlier that we have a very responsive higher education curriculum, graduates from that degree program, specifically agricultural biotechnology, tends to work on regulatory agencies in the Philippines. And that's why the regulatory bodies are capable enough to handle different kinds of regulatory processes. And another good thing about the biotech systems in the Philippines is that regulation of biotech crops are independently assessed by qualified professionals. And regulatory system is relevant or relatively science-based and evidence-based, and it's case by case. We don't have a blanket, definition of things. So that's why things are kind of moving more fluidly in terms of different products that are being applied in the regulatory system. And lastly, we have a progressive outlook on gene-edited crops. As I mentioned, we are not regulating it like GMOs. So what's the downside or the challenges on the government and regulatory sector? So there's a language mismatch between scientists and the judiciary department. So since the government is also, since the judiciary is also part of the government, we should also engage them, especially in times when, anti-GM groups are suing us. So there's a mismatch between the judiciary and the scientific community, because when the judiciary asks the scientists, is it safe? The scientists cannot say that it's 100% safe. So there's a language mismatch between the scientists and the judiciary. And of course, the regulatory system is still very complex and a lengthy process in terms of how it is being done in the Philippines, although relatively it has improved from the previous versions of the regulatory framework. And there's a lack of comprehensive national biotechnology roadmap for the biotech crops in the Philippines. It's not so coordinated as we wanted to be as scientists or researchers in the Philippines. But hopefully in the next coming years, there's going to be more developments and improvements in terms of the roadmaps that is being applied in the biotech sector in the Philippines. Let's now move on to the best practices and the end users portion of the sector, and also the civil society sector. So there's an extensive network of biotech advocates. I, for one, was first exposed to biotechnology as a volunteer in a youth organization, and that explains a lot in terms of how extensive the biotech advocate space in the Philippines is. And also, public participation in the regulatory process is guaranteed. So the regulatory framework guarantees public participation, and there's also safeguards in place to allow the public to scrutinize certain decisions, whether it is positive or negative in terms of approval process. The public can demand to petition to the regulatory bodies. So there are safeguards. And lastly, there are media resources. There's a lot of pool of media resources that are available in internet, in the web for the Philippine side. But we also face these things similarly to a lot of countries here. So there's a highly organized network of anti-GM groups stemming from other wealthier countries. And because of that, they abuse the use of administrative and legal remedies in the regulatory process, and that delays the processes even more. And the communication efforts in biotech crops are done by tech developers as well. So tech developers have a lot of things on their plate, and they have to still, communicate the science behind it. And so it gives them more work and less time to do research in this space. So here are my key takeaways. So there's no silver bullet. So replicating one good thing and overcoming one bad thing will not automatically result to a perfect biotech system. So there must be concerted efforts by all stakeholders in order to create enabling environments for biotech innovations to thrive. So by presenting this biotech systems of the Philippines, I hope I was able to inspire you guys into thinking into systems. So first, we should evaluate our existing systems. And after evaluating the systems that we have, we can now mobilize to solve the problems in the systems. And lastly, we can then transform the agricultural sector, not just in Africa, but in the entire global south. Thank you very much.
Thank you, Albert. So we're about to wrap up. We're left with just one more thing. We are doing some consultation on how to format the last part so that it's done. So you are good to go? Fantastic. All right, so let's do this quickly. Just rise to your feet. You've been sitting for too long. So while you stretch, we'll get a table set for the last part. Just stretch around, just move around. This is the last. It's Friday, Please, guys, set the tables for us. Talk to somebody. Shake hands with somebody. Say something to anybody. I mean, just do something. Let's do this for 2 minutes. All right. Thank you. Please, let's take our seats. Kindly take your seats. We want to wrap up today. Everything we've heard from morning through the afternoon to this time. What is the conclusion of the whole matter? It is about the future of Africa's biotech crops. That is going to be the future of the whole matter. Now we are going to invite the following upstage, and each of them will have one opportunity to make a statement for 3 minutes. Just 3 minutes, not more. There are 3 people we haven't heard from in the entire program. Those will give 3.5 minutes to, the rest 3 minutes. So we haven't heard from Dr. John Eleblu today, we have not heard from Dr. Michael Osai today, and we haven't heard from Professor Kwame Ofe. So those will have 3.5 minutes. The rest will have 2 minutes, 3 minutes each. So Dr. Murenga, Dr. Elizabeth Nuyunga, Dr. John Udipio, Dr. John Eleblu, Dr. Michael Osai, please come up stage. We are wrapping up the entire discussion. What is the takeaway? What are we going away with regarding the future of Africa's biotech crops? Please, this session will be moderated by The session chair, Professor Kwame Ofe, Director, Biotechnology Center, University of Ghana. Prof, please. Each of them has only 3 minutes.
So each of them would just respond to the same question. What is the future of biotechnology? So my work has been made very simple. No introduction. I just go straight and pose the questions to them very easily. Okay, so where do I start from? I should start from there.
Anyway,
very well. Okay. So what is your perspective on the future of biotechnology in Africa? You have 3 minutes each? Or 3 minutes each. Okay, so who's timing? Or myself?
You go first. Ladies first.
Okay, so gentleman on the extreme left.
All right. Thank you very much, and good afternoon, audience. I believe with the challenges we have with climate change, the increasing population, and the need to feed a lot more mouths, it is important that we increase productivity. It is important that we increase the resilience of our crops. It is important that we increase the nutritional benefits from our crops. So if we are looking at the future of biotech, these are the areas we should focus. How are we going to make our crops more resilient? And this, I mean our indigenous crop varieties. How will we make them more resilient to climate change? How will we make them more resilient to climate-driven pests and diseases? Because these are posing a lot of challenges to our agriculture. How are we going to increase the nutritional benefits that we derive from the foods that we are producing? If we are going to incorporate biofortification into our breeding systems, that would benefit our populations. How are we dealing with the issues that challenge our food systems? So if we find that we have been using conventional methods to do this or to solve these problems, but we haven't gone very far. If you look at the African continent, for almost every crop, our level of productivity is the lowest. So we will need to find solutions to improving productivity. And biotech or modern biotechnology comes with that solution. It is possible that with modern biotechnology, we can actually even stack different traits into the same crop to make it such that the crop is tolerant to all the climate stresses and at the same time high yielding and at the same time giving us superior nutrition. So if we rely on this technology, we can solve most of our challenges that face our food systems. But to do this, we require that we set proper regulatory systems in place. In Ghana, at least we have our national biosafety system in place, and we are working hard to make sure that we also get regulations for the new biotechnology systems that are coming up for crop breeding. So I think that is the future of biotech, to be open-minded and allow the systems to regulate the biotechnology for us to be able to derive the benefits from it. Thank you.
Thank you very much. Our next panelist.
Thank you. After my colleague here has spoken, I don't know if there's much more to add to it. Maybe I'll repeat in my own words. But I must say, after the presentations today, I'm very well encouraged, and I feel like there's already so much that is happening in Africa. It's not as gloomy as it may appear. There's already so many things on pipeline. There's products that are almost there, and also the regulatory processes that are already becoming a bit more friendly. When I hear Kenya and Nigeria, we're already looking at GE in a different way as opposed to GMO. So I'm quite well convinced that the future can only be brighter. Biotech have a bright future. as we look to climate change adaptation. I think see and tell will be the solution for us, that when we plant in some of these plants in field trials or environmental release, and the people come and see. I think this has happened before in other previous technologies, that they were initially rejected, but then when you see the outcome and the good part of it, then it will be accepted. The future is bright for biotech crops. Yeah.
Thank you very much. The future is bright for biotechnology crops. Thank you. Next.
Yes, thank you for giving me this opportunity to give my perspective. Some of the things I have to say have already been said, but most importantly, I think we are heading in the right direction, where in Africa, biotechnology is not going to be totally forgotten about. Already with the help of donor participation, capacity building has started in almost all areas of biotechnology and plant breeding in general. In an institution like this where we are gathered, there are students graduating almost every year, and in other universities, and also from national research institutions, there are also people being trained to venture into biotechnology. That is the base of biotechnology. So when we have people trained like that, and then we back them with apportioning significant amount of resources so that they can explore research in the area of biotechnology, and then we can get products and services that will be for Africa, made by Africans. But as we do all this, I see awareness in terms of regulation already being created. So should we go in the right direction to create science-based frameworks and laws for the products and services derived from biotechnology, then I see a case where we shall have climate-smart products which combine high level of nutrition and the resilience to a number of constraints in the world, which will be out there for our farmers in Africa. And so I really feel that it's just a matter of time before we realize these benefits. But for that to happen, we definitely have to ensure that we invest in it in order to realize the goodness of biotechnology that we can get in Africa. Thank you.
Thank you very much. So building capacity, creation of awareness, engaging regulators. Thank you.
Yeah. Prof, thank you for the question, and audience. I came with a lot of write-up to deliver a powerful speech. But I have only 3 minutes. Anyway, I'll make it short. The scenario in Africa is not we are not there yet. I wouldn't say that we are doing well. We have a lot to do when you look at the food security and nutritional deficiencies that we have. Actually, at this conference, we should also have national security and policy makers and all those guys that need to drive the system for us to move quickly. Because looking at the current scenario, where you have crops being edited with all the biotech tools and amazing things happening, even able to edit the epigenome, that's epigenetic regulation, beautiful things can be done, but the system is not, in Africa, is not ready to take up these innovations quickly and get it to farmers. In Ghana, we've gone through a lot, but we've just approved one event, and it's not even an edited event. is a GMO that has been approved, but then we want to go through the processes of testing over and over and over again, which I believe is quite limiting, and it slows down the pace at which we get varieties and seeds to farmers. And whilst we are doing this, a lot of people are suffering and are dying, and I believe that we need to look at better ways of going about regulating gene-edited crops especially. If you look at the case of Japan, where they've already gone through approvals for 4 events, edited events, and they treat them as non-GM because they've looked at it and they see that there is no foreign material, and so quickly it goes out. We should look at such examples and learn from Japan, not learning from others that have been very, very slow and also against progress. Africa is at the point where we need to emerge as leaders, and we should adopt all the technologies that are in the toolbox. And Dr. Alina is doing wonderful work that must be pushed, and this cannot be done and then to be on the shelves as usual and not get to farmers. We really need to move quickly, and that's all I have to say. Thank you.
Thank you very much. I was distracted, so I didn't pay attention, but I agree with you. Yes, we have a little bit more time, and I want us just to consider one more question. What do you think should be our priorities in biotechnology in Africa? General. Yes.
I think our prime focus should be to look at our staples, our staple crops, and see how we can adopt this biotechnology to improve those staple crops for the benefit of our people.
May I also add that we need to streamline our regulatory systems. We can't just have 2 countries for the GE that are looking at it as non-GMO. Out of the 54 countries, there's no statistics there. So regulatory barriers have cost us a lot of challenge. At least I can speak for my PhD that it took much longer and there was a lot of challenges therein. And many more others who will look for funding and will not get it because what are you going to do with the funding that is, I mean, a product that is not going to be marketed in your own country? Why would donors want to put in their money there? So with all the products again that are coming and looking at them, surely we need a supportive regulatory system.
Supportive regulatory systems. Yes, indeed. Colleague.
Yes, for me, I would say we have to decide who should drive our development agenda. Right now, we seem to be our countries, some of our countries, they seem to be easily convinced to leave things which may be of interest for their development because another partner has said, don't do this, not knowing that the interest of the one telling you might be different. And if we know our development agenda, then we will be able to stand for it. And as we stand for it, we will be able to invest in it in order to realize what we want as a country. So we first have to know what we want, and then we will go to how we can achieve it.
So we should stand on our feet and be focused on our own development agenda, not that by the one who is funding. There's a saying that he who calls the pays the piper calls the tune. You know about that saying, though. Okay, John.
The top priority should be capacity building and quick adoption. Quick adoption of technology, just like we adopt mobile phones and all the good gadgets in Africa. In fact, we lead in adoption of mobile phones. And I think it's important that we lead in adoption of technologies for food consumption.
Thank you very much. Thank you very much. On that note, I think we'll bring this session to an end. And I'll invite Abigail to come and give us the summary. Is she here? Yes. Thank you very much.
Good evening. It's been a long day, I can see, and also soon this insightful symposium is coming to a closure. To keep us all on the same page, I would want to share a few pointers. And I believe this will generate future discussions as well as contribute to increasing access to new technologies in Africa. Throughout the day, all the presentations we saw or heard made mention of a number of issues, and they ran through a number of them. One among which is the creation of awareness. I would say that it's become very important that Africa invest in the creation of awareness on existing technologies that are available in Africa. And to do this, I know that the media plays a very key role here. In order to disseminate accurate information, we would want to urge the media to do a good job in disseminating a very accurate information to allay the fear of the public. Again, throughout the day, we've heard that, or we have seen that the map of Africa is not looking green. In order for it to look green, we have to deploy interdisciplinary approach, which include paying attention to political and then the social environment, so that we can see or we can have the Africa we want. In one of the presentations, we got to know that the economy of Africa can be improved if we adopt this technology as soon as possible. And in order to do this, we need an enabling environment. Again, the map of Africa has to be improved. We realize that when it comes to genome editing, only 3 African countries have functional guidelines in place. We urge all Africa to take the bold step in ensuring that there are functional regulatory systems in place so that we can increase the human capacity and also have our scientists be able to produce products that will reach the farmer and at large reach the consumer as well. Last but not least, I would want to add that in order for us to see the Africa we want, although we welcome donor funding, we want to say that Africa must arise and invest more in research and development to see the Africa we want. In doing this, we urge each and every one to be part of the process. In doing this, we urge each and every one to take the bold step to advocate for technologies like this to see the Africa we want. Thank you very much.
Now the day is over. Night is drawing nigh. Shadows of the evening still across the sky. You've all gone to Sunday school. Good one. Put your hands together for yourselves. Okay, thank you very much, everyone, for making this possible. We are taking the closing remarks, final closing remarks from Professor Kwame Ofe, who is the director of Biotechnology Center, University of Ghana. And when Professor is done and you don't see me up here again, my name is Chris Wala Esikpe. It's been my pleasure doing this with you. Alumni, please stay behind when we are done. Thank you very much. Let's do this again and again and again.
Thank you very much. Ladies and gentlemen, throughout our discussions, our scientists developing improved crop varieties using these advanced technologies have showcased the tremendous potential they hold for transforming agriculture in Africa. The insights shared by the scientists developing improved varieties of cassava, banana, cowpea, and cotton using these new breeding techniques have provided us with a glimpse into the realms of possibilities that can offer solutions to address food security, climate resilience, and nutrition challenges facing our continent. It is, however, crucial to address the challenges that have inhibited the rapid adoption of these technologies in Africa. Misconceptions surrounding GMOs and gene editing have perpetuated apprehension and skepticism among the public. During this symposium, we have shed light on the scientific evidence, safety protocols, and regulatory frameworks that govern these technologies. By fostering open and transparent dialogue, we can dispel these misconceptions and foster public trust and avoid the backlash experienced in other continents when these technologies were introduced. We need to initiate a collaboration between scientists, government officials, and regulatory bodies to develop regulatory frameworks that embrace scientific advancements with safeguarding public health and environmental integrity. Let us avoid the language of mismatch between scientists and regulators and work together towards a common goal. By addressing concerns related to access, affordability, and ownership, biotech crops, we can ensure that the benefits reach those who need them the most, fostering inclusive and sustainable agricultural development. As we move forward, let us continue to engage in informed conversations, promote evidence-based decision-making, work collaboratively to address the challenges that have hindered the rapid adoption and promotion of biotechnologies in Africa. Let us embrace the transformative potential of GMOs and gene editing to avoid and overcome food insecurity, promote resilience, and unlock new opportunities for economic growth and social progress. I thank you all for your invaluable contributions, and I wish you safe travels on your journey home. Together, let us shape the future of biotechnology in Africa. I thank you all very much. So should I say that is the end then? Thank you all once again. God bless you.
side